50 share. Cool. All right. Let's do it. See. Yeah. Okay. Cool.
With a glucose molecule. We start off with a glucose molecule, which is a 6-carbon molecule.
I'll go back. Let me wash my face. Look for videos that explain the cycle first. Yeah, I did some reading about it, so well, I just did some pre-reading because I'm taking physiology, so I have some understanding of it now. And then I think Yusuf still has his videos too. I think he has it in. Actually, this is probably the order I'm going to do on the next parts. I think he probably has it here, right? Yeah, right here. Yep. He's got them all right here at the end. So we're gonna. Oh, you guys can't even see what I'm seeing, huh? This guy. He has it as well. Yeah, there's probably a lot of videos on YouTube, but people recommend to watch his videos, so I'll do that. I'll do that. Biochem, and then I'll probably take some time, like some time while I'm doing Gen Chem, and do the practice questions, the question banks. I can get started on that. So like what I'll do is I mean I don't know. Maybe I might just do it on Share. Okay. I'll do these ones here. Wait, is it these ones? This one? I don't know. One of these. I'll do them. No, where was it? Oh, this one. This one. Sorry. I'm gonna do these two, and this one. I don't know. As many of these as I can do. Like get rid of these, um, because my test is coming up pretty soon, so might as well. Might as well. Might as well. Might as well. All right. Let me wash my face. Happy. Oh, thanks, Happy. I appreciate it, buddy. Thank you so much. Thanks, Happy. Appreciate it, buddy. Heh, lol. Just wrote an exam about like. Just wrote an exam about that like 2 weeks ago. Okay. Yeah. Thanks. All right. Let me wash my face and put on some eye drops. I'll be back. And oh, that feels great. Yep. It's a little more on the right side. That's good stuff.
Six carbon molecule. This essentially gets split in half by glycolysis, and we end up with two pyruvic acids or two pyruvate molecules. So glycolysis literally splits this in half. It lyses the glucose, and we end up with two pyruvates or pyruvic acids. Pyruvate. And these are three carbon molecules. Three carbon molecules. There's obviously a lot of other stuff going on in the carbon you saw in the past, and you can look up their chemical structures on the internet or Wikipedia and see them in detail. But this is kind of the important thing: is that it was lysed, it was cut in half, and this is what happened in glycolysis. 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 And this happened in the absence of oxygen, or not necessarily. It can happen in the presence or in the absence of oxygen. It doesn't need oxygen. And we got a net payoff of 2 ATPs net. And I always say the net there because remember, it used 2 ATPs in kind of that investment stage, and then it generated 4. So on a net basis, it generated 4, used 2, it gave us 2 ATPs, and it also produced 2 NADHs. 2 NADHs. That's what we got out of glycolysis. And this is a little bit better. Let me draw a cell right here. Maybe I'll draw it down here. Let's say I have a cell that's its outer membrane. Maybe it's a nucleus. We're dealing with a eukaryotic cell. It doesn't have to be the case. It has this DNA that's chromatin form all spread around like that. And then you have some mitochondria. And there's a reason why people call it the power centers of the cell. We'll look at that in a second. So there's a mitochondria, has a membrane and inner membrane just like that. I'll do more detail on the structure of a mitochondria maybe later in this video, or maybe I'll do a whole video on them. It's another mitochondria right there. And then all of this fluidic space out here that's between the organelles, where the organelles, you can view them as parts of the cell that are, they do specific things. They do specific things, kind of like organs do specific things within our own bodies. So between all of the organelles, you have just this fluidic space. This is just fluid of the cell, and that's called the cytoplasm. Cytoplasm. And that's where glycolysis occurs. So glycolysis occurs in the cytoplasm. Glycolysis. Now, we all know, in the overview video, we know what the next step is. It's the Krebs cycle, or the citric acid cycle. And that actually takes place in the inner membrane, or the inner space of these mitochondria. Let me draw it a little bit bigger. Let me draw mitochondria here. So if this is a mitochondria, it has an outer membrane. It has an inner membrane. If I have just one inner membrane, we call it a crista. If we have many, we call them cristae. These little convoluted inner membrane. Give it a label. So they're cristae, plural. And then it has 2 compartments, right? It's because it's divided by these 2 membranes. This compartment right here is called the outer compartment. This whole thing right there, that's the outer compartment. And then this inner compartment in here is called the matrix. The inner compartment right there is called the matrix. Now, you have these pyruvates. They're not quite just ready for the Krebs cycle, but I guess, well, it's a good intro to how do you make them ready for the Krebs cycle? They actually get oxidized. And I'll just focus on one of these pyruvates. We just have to remember that the pyruvate, that this happens twice for every molecule of glucose. So we have this kind of preparation step for the Krebs cycle. We call that pyruvate oxidation. Pyruvate oxidation. And essentially what it does is it cleaves one of these carbons off of the pyruvate. And so you end up with a 2-carbon compound. You don't have just 2 carbons, but its backbone of carbons is just 2 carbons, called acetyl-CoA. Acetyl-CoA. And if these names are confusing, because what's acetyl coenzyme A, these are very bizarre, you could do a web search on them. But I'm just going to use the words right now, so we'll keep things simple and look at the big picture. So it generates acetyl-CoA, which is this 2-carbon compound. And it also reduces some NAD plus to NADH. And this process right here is often given credit, or the Krebs cycle, the citric acid cycle, gets credit for this step, but it's really a preparation step for the Krebs cycle. Now, once you have this 2-carbon chain acetyl-CoA right here, you are ready to jump into the Krebs cycle, this long talked about Krebs cycle. And you'll see in a second why it's called the cycle. The acetyl-CoA, and all of this is catalyzed by enzymes. And enzymes are just proteins that bring together the constituent things that need to react in the right way so that they do react. So catalyzed by enzymes, this acetyl-CoA merges with some oxaloacetic acid, very fancy word, but this is a 4-carbon molecule. This is a 4-carbon molecule right here. These 2 guys are kind of reacted together or merged together, depending on how you want to view it. Drop like that. All catalyzed by enzymes. It's important, some tests will say, is this an enzyme catalyzed reaction? Yes, everything in the Krebs cycle is an enzyme catalyzed reaction, and they form citrate or citric acid. Citric acid, which is the same stuff in your lemonade or your orange juice. And this is a 6-carbon molecule, which makes sense. You have a 2-carbon and a 4-carbon. You get a 6-carbon molecule. And then the citric acid is then oxidized over a bunch of steps. And this is a huge simplification here, but it's just oxidized over a bunch of steps. Again, the carbons are cleaved off. Both 2 carbons are cleaved off of it to get back to oxaloacetic acid. And you might be saying, hey, when these carbons are cleaved off, like, when, let's say this carbon is cleaved off, what happens to it? It becomes CO2. It gets put onto some oxygen and leaves the system. So this is where the oxygen or the carbons or the carbon dioxide actually gets formed. And similarly, when these carbons get cleaved off, it forms CO2. And actually, for every molecule of glucose, you have 6 carbons. when you do this whole process once, you're generating 3 molecules of carbon dioxide, but you're going to do it twice. You're going to have 6 carbon dioxides produced, which accounts for all of the carbons, right? You get rid of 3 carbons for every turn of this, well, 2 for every turn, but really for the steps after glycolysis, you get rid of 3 carbons, but you're going to do it for each of the pyruvates. You're going to get rid of all 6 carbons, which will have to exhale eventually. But this cycle, it doesn't just generate carbons. The whole idea is to generate NADHs and FADH2s and ATPs. So, right here, and this is a huge simplification. I'll show you the detailed picture in a second. We'll reduce some NAD into NADH, NAD plus into NADH. We'll do it again. And of course, these are in separate steps. There's intermediate compounds. I'll show you those in a second. NAD plus, another NAD plus molecule will be reduced to NADH. It will produce some ATP will turn into, some ADP will turn into ATP. ADP turning into ATP. Maybe we have some, not maybe, this is what happens. Some FAD gets, let me write it this way. Some FAD gets oxidized into FADH2. And the whole reason why we even pay attention to these, you might think, hey, cellular respiration is all about ATP. Why do we even pay attention to these NADHs and these FADH2s that get produced as part of the process? The reason why we care is that these are the inputs into the electron transport chain. These get oxidized, or they lose their hydrogen in the electron transport chain, and that's where the bulk of the ATP is actually produced. And then maybe we'll have another NAD get reduced or gain in hydrogen, right? Reduction is gaining an electron or gaining a hydrogen whose electron you can hog. NADH, and then we end up back at oxaloacetic acid, and we can perform the whole citric acid cycle over again. So now that we've written it all out, let's account for what we have. So depending on, let me draw some dividing lines so we know what's what. So this right here, everything to the left of that line right there is glycolysis. Glycolysis, we learned that already. And then most, especially introductory textbooks, will give the Krebs cycle credit for this pyruvate oxidation, but that's really a preparatory stage. The Krebs cycle is really formally this part where you start with acetyl-CoA, you merge it with oxaloacetic acid, and then you go and you form citric acid, and which essentially gets oxidized and produces all of these things that we need to either directly produce ATP or you do it indirectly in the electron transport chain. But let's account for everything that we have. Let's account for everything that we have so far. We already accounted for the glycolysis right there, 2 net ATPs, 2 NADHs. Now, in the citric acid cycle, or in the Krebs cycle, well, first we have our pyruvate oxidation that produced 1 NADH. But remember, if we want to say what are we producing for every glucose, this is what we produce for each of the pyruvates, right? This was from 1 NADH from just this pyruvate. But glycolysis produced 2 pyruvates. So everything after this, we're going to multiply by 2 for every molecule of glucose. So I'll say, so the pyruvate oxidation times 2 means that we got 2 NADHs, 2 NADHs right there. And then when we look at this side, when we have the formal Krebs cycle, what do we get? We have how many NADHs? 1, 2, 3 NADHs. So 3 NADHs times 2, because we're going to perform this cycle for each of the pyruvates produced from glycolysis. So that gives us 6 NADHs. We have 1 ATP per turn of the cycle. It's going to happen twice, once for each pyruvic acid. So we get 2 ATPs. And then we have 1 FADH2. We're going to do this cycle twice. This is per cycle, so times 2. We have 2 FADHs. Now, sometimes in a lot of books, these 2 NADHs, or per turn of the Krebs cycle, or per pyruvate with this 1 NADH, they'll give credit to the Krebs cycle for that. So sometimes instead of having this intermediate step, they'll just write 4 NADHs right here, and you're going to do it twice, once for each pyruvate. So they'll say 8 NADHs get produced from the Krebs cycle. But the reality is 6 from the Krebs cycle, 2 from the preparatory stage. Now, the interesting thing is we can now count whether we get to the 38 ATPs promised by cellular respiration. We've directly already produced for every molecule of glucose 2 ATPs and then 2 more ATPs. So we have 4 ATPs. 4 ATPs. How many NADHs do we have? 2, 4, and then 4 plus 6 is 10. We have 10 NADHs. And then we have 2 FADHs, 2 FADH2s. I think in the first video on cellular respiration, I said FADH. It should be FADH2, just to be particular about things. And these, so you might say, hey, where's our 38 ATPs? We only have 4 ATPs right now. But these are actually the inputs in the electron transport chain. These molecules right here get oxidized in the electron transport chain. Every NADH in the electron transport chain produces 3 ATPs. So these 10 NADHs are going to produce 30 ATPs in the electron transport chain. And each FADH2, when it gets oxidized and gets turned back into FAD in the electron transport chain, will produce 2 ATPs. So 2 of them are going to produce 4 ATPs in the electron transport chain. So we now see we get 4 from just what we've done so far, glycolysis, the preparatory stage, and the Krebs or citric acid cycle. And then eventually these outputs from glycolysis and the citric acid cycle, when they get into the electron transport chain, are going to produce another 34. So 34 plus 4, it does get us to the promised 38 ATP that you would expect in a super efficient cell. This is kind of theoretical maximum. In most cells, they really don't get quite there, but this is a good number to know if you're going to take the AP Bio test or in most introductory biology courses. There's one other point I want to make here. Everything we've talked about so far, this is carbohydrate metabolism, or sugar catabolism, we could call it. We're breaking down sugars to produce ATP. Glucose was our starting point. But animals, including us, we can catabolize other things. We can catabolize proteins. We can catabolize proteins. We can catabolize fats. And if you have any fat on your body, you have energy. You're in theory, your body should be able to take that fat and you should be able to do things with that. You should be able to generate ATP. And the interesting thing, the reason why I bring it up here is obviously glycolysis is of no use to these things, although fats can be turned into glucose in the liver. But what the interesting thing is that the Krebs cycle is the entry point for these other catabolic mechanisms. Proteins can be broken down into amino acids, which can be broken down into acetyl-CoA. Fat can be turned into glucose, which actually could then go the whole cellular respiration. But the big picture here is acetyl-CoA is the general catabolic kind of intermediary that can then enter the Krebs cycle and generate ATP regardless of whether our fuel is carbohydrates, sugars, proteins, or fats. Now, we have a good sense of how everything works out right now, I think. Now I'm going to show you kind of a diagram that you might see in your biology textbook, or I'll actually show you the actual diagram from Wikipedia. And I just want to show you this looks very daunting and very confusing, and I think that's why many of us have trouble with cellular respiration initially, because there's just so much information. It's hard to process what's important. But I want to just highlight the important steps here, just so you see it's the same thing that we talked about. From glycolysis, you produce 2 pyruvates. That's the pyruvate right there. That actually shows molecular structure. This is the pyruvate oxidation step that I talked about, the preparatory step. And you see we produce a carbon dioxide, and we reduce an NAD plus into NADH. Then we're ready to enter the Krebs cycle. The acetyl-CoA and the oxaloacetate, or oxaloacetic acid, they are reacted together to create citric acid. They've actually drawn the molecule there. And then the citric acid is oxidized through the Krebs cycle right there. There's all of these steps. Each of these steps are facilitated by enzymes, and it gets oxidized. But I want to highlight the interesting parts. Here, we have an NAD get reduced to NADH. We have another NAD get reduced to NADH. And then over here, another NAD get reduced to NADH. So far, if you include the preparatory step, we've had 4 NADHs formed, 3 directly from the Krebs cycle. That's just what I told you. Now we have, in this diagram, they say GDP or GTP gets formed from GDP. The GTP is just guanosine triphosphate. It's another purine that can be a source of energy, but then that later can be used to form an ATP. So this is just the way they happen to draw it, but this is the actual ATP that I drew in the diagram off the top. And then they have this Q group, and I won't go into it, and then it gets reduced over here. It gets those 2 hydrogens, but that essentially ends up reducing the FADH2s. So this is where the FADH2 gets produced. So as promised, we produced for each pyruvate that inputted, remember, so we're going to do it twice. For each pyruvate, we produced 1, 2, 3, 4 NADHs. We produced 1 ATP and 1 FADH2. That's exactly what we saw up here. I'll see you in the next video.
Okay, hold on, let me get my jacket.
Before talking about the regulation that occurs inside the citric acid cycle, let's take a moment and step back and talk about what regulates the entry into the citric acid cycle. So remember that a molecule called acetyl-CoA is what really enters the citric acid cycle and is oxidized into the carbon dioxide molecules as it kind of goes around in the citric acid cycle. And instead of writing out the entire chemical formula, I just want to abbreviate this as a 2-carbon molecule with the coenzyme A functional group, which is actually a thiol group, a sulfur group. So I'll just write 2 carbons with a sulfur coenzyme group for short. Now I want to remind you what produces acetyl-CoA. So remember, we have glycolysis. And from glycolysis, which begins the breakdown of glucose, we produce pyruvate. And so it's the pyruvate that travels from the cytosol into the mitochondria that's converted into acetyl-CoA by a very special enzyme called pyruvate dehydrogenase. And remember, dehydrogenase means we're dehydrogenating or oxidizing our molecule. And so if we're oxidizing, it shouldn't surprise you then that this enzyme has a cofactor. Indeed, it requires NAD+, which is converted into NADH, or I should say reduced into NADH, as pyruvate is being oxidized into acetyl-CoA. And I want to remind you that pyruvate is a 3-carbon molecule, so it's losing a carbon molecule. You can see here, because acetyl-CoA is 2 carbons, but pyruvate is 3, so a carbon must be lost during this reaction. And indeed, part of the oxidation process releases a carbon dioxide molecule. And finally, I also want to note as well that, of course, in order to get this coenzyme A here, we need to have that as a substrate as well. Now, one important point about this step, this entry point into the citric acid cycle, is that this reaction in going from pyruvate to acetyl-CoA is irreversible, which is why I'm kind of bolding this unidirectional arrow here to tell you that while we can take pyruvate into acetyl-CoA, it's not urban because acetyl-CoA, as pyruvate is being oxidized into acetyl-CoA. And I want to remind you that pyruvate is a 3-carbon molecule, so it's losing a carbon molecule. You can see here, because acetyl-CoA is 2 carbons, but pyruvate is 3, so a carbon must be lost during this reaction. And indeed, part of the oxidation process releases a carbon dioxide molecule. And finally, I also want to note as well that, of course, in order to get this coenzyme A here, we need to have that as a substrate as well. Now, one important point about this step, this entry point into the citric acid cycle, is that this reaction in going from pyruvate to acetyl-CoA is irreversible, which is why I'm kind of bolding this unidirectional arrow here to tell you that while we can take pyruvate into acetyl-CoA, it's not possible to take acetyl-CoA and turn it into pyruvate. And remember that when we say a reaction is irreversible, that's just another way to say that we have a pretty large negative delta G value. Now, the big point I want to make is that because this reaction is irreversible, it makes it a nice target for the cell for lots of regulation. And remember that regulation often occurs on irreversible steps because these are the steps that if you open, basically, then the ball will keep rolling down the pathway. So we want to make sure that these irreversible steps are tightly regulated. But just as a quick side note, before we talk about the actual regulation, it's also kind of nice to recognize that fatty acids can also contribute to the production of acetyl-CoA when they're broken down, but because this reaction is irreversible, this acetyl-CoA produced by fatty acids cannot contribute to the production of pyruvate and therefore cannot contribute to the production of gluconeogenesis, which, remember, if you recall, occurs by using pyruvate as one of the substrates. And so that's why you might hear some textbooks kind of quote this fact that fatty acids, or at least most of them, cannot contribute to the production of glucose. All right, so let's leave that tangent for a moment and let's return to our question, which is how is the production of acetyl-CoA regulated? And to answer this question, I first have to kind of just start off with the big picture, which is what is the purpose of acetyl-CoA? And the two major purposes are one, which most people are familiar with, which is entry into the citric acid cycle. And of course, the entry into the citric acid cycle allows the acetyl-CoA to be oxidized into carbon dioxide and produce the electron carrier molecules NADH and FADH2, which then enter the electron transport chain to produce ATP. All right, so that's one purpose. But another purpose is also, remember how I mentioned that fatty acids can be broken down to acetyl-CoA? Well, acetyl-CoA can also be used to produce fatty acids when ATP levels are high. And so this is kind of the second major use of acetyl-CoA in the body. And so keep these kind of two major pathways for acetyl-CoA in mind as we talk about how this step is regulated. And I should say at this point that the major form of regulation in this case is allosteric regulation of the pyruvate dehydrogenase enzyme. So remember, that's just a fancy way for saying that there are molecules that can essentially bind to a part of the enzyme to make it work better, in which case it's an allosteric activator, or to make it not work as good, in which case it would be an allosteric inhibitor. And I kind of remember that this is a main form of regulation in this step because it really allows this step to kind of assess the energy state of the body by looking at kind of what molecules it has floating around. And in fact, let's go ahead and write out what some of the allosteric activators and inhibitors are of this pyruvate dehydrogenase enzyme, and you'll kind of see what I mean when I'm talking about the energy state of the cell. Now, one important principle that I use to kind of remind myself what activates and inhibits this enzyme is to remember what the products and the substrates are for this reaction. And I kind of essentially think back to Le Chatelier's principle and just say to myself that if we have an accumulation of substrates, these are going to want to be allosteric activators. Essentially, they want to push this kind of reaction forward. And if we have an accumulation of the products, these are going to probably most likely be allosteric inhibitors because they're going to, assign that too much is being produced and we can put a brake on the reaction. And so indeed, the allosteric activators include the substrates CoA, as well as NAD+, and even pyruvate. And then the inhibitors include acetyl-CoA, of course, as well as NADH. Now, a couple more allosteric activators and inhibitors that might not be immediately obvious, but will make sense once we discuss them are ATP is also a negative allosteric inhibitor. On the flip side, AMP is an allosteric activator. And additionally, fatty acids can also be an additional allosteric inhibitor, and calcium can be an additional allosteric activator. All right, so how can we reset these final allosteric activators and inhibitors? Well, first, simply realize that the levels of ATP and AMP, again, are getting at this energy state of the cell. It's a way for the body to assess if it needs to shuttle more acetyl-CoA through the citric acid cycle and then, channel all of these electron carrier molecules to the electron transfer chain, or whether it has enough ATP and it can slow down the flux of acetyl-CoA through the citric acid cycle. And so it should make sense to you that having a low energy state in the cell, indicated by lots of AMP, should activate this, should alert the body to produce more acetyl-CoA. But if we have enough ATP lying around, then this reaction can essentially slow down. Now, this calcium here may not be immediately obvious, but I'll remind you that remember that exercising skeletal muscle involves the influx of a lot of calcium. And so when you're exercising, of course, energy needs go activator.
Yeah, okay.
Additionally, fatty acids and even pyruvate. And then the inhibitors include acetyl-CoA, of course, as well as NADH. Now, a couple more allosteric activators and inhibitors that might not be immediately obvious, but will make sense once we discuss them, are ATP is also a negative allosteric inhibitor. On the flip side, AMP is an allosteric activator. And additionally, fatty acids can also be an additional allosteric inhibitor, and calcium can be an additional allosteric activator. All right, so how can we reason out these final allosteric activators and inhibitors? Well, first, simply realize that the levels of ATP and AMP, again, are getting at this energy state of the cell. It's a way for the body to assess if it needs to shuttle more acetyl-CoA through the citric acid cycle and then, channel all of these electron carrier molecules to the electron transfer chain, or whether it has enough ATP and it can slow down the flux of acetyl-CoA through the citric acid cycle. And so it should make sense to you that having a low energy state in the cell, indicated by lots of AMP, should activate this, should alert the body to produce more acetyl-CoA. But if we have enough ATP lying around, then this reaction can essentially slow down. Now, this calcium here may not be immediately obvious, so I'll remind you that remember that exercising skeletal muscle involves the influx of a lot of calcium. And so when you're exercising, of course, your energy needs go up. And so in skeletal muscle, this free calcium in the cell is kind of a nice alert to say, you know what, we're going to need more energy. Let's start producing more acetyl-CoA. And finally, these fatty acids. Why do these fatty acids inhibit the production of acetyl-CoA? Well, of course, I remind you here of the second purpose of acetyl-CoA, which is to produce the synthesis of fatty acids. And so if we have enough fatty acids in the body, again, it can be a signal to the cell to say, you know what, we don't need any more acetyl-CoA. We can slow this process down. So at the end of the day, there might be a lot of these allosteric regulators to keep track of, but just go back to the basics and remind yourself what is a substrate, what is a product, and what the energy state of the cell is. And I think you'll be able to reason out most of these. To save us some time, I've went ahead and drawn out a simplified version of the citric acid cycle here. And if you remember, it begins with acetyl-CoA entering the cycle, and it combines with this molecule oxaloacetate to form citrate. And this citrate undergoes various conversions and oxidations, which eventually cause the two carbons that entered with acetyl-CoA, because remember, acetyl-CoA is a 2-carbon molecule. These 2 carbons exit as carbon dioxide. And as it's oxidized and loses its carbon dioxide, acetyl-CoA also allows us to produce the electron carrier molecules FADH and NADH, which, if you recall, go to the electron transfer chain to allow us to produce ATP. So to summarize, let's go ahead and write out the kind of overall chemical reaction of the citric acid cycle. So we have as our reactants acetyl-CoA entering the cycle. We also have some coenzymes like NAD+ and FAD. And we also have a free GDP. Remember, GTP is formed in this cycle as well. And all of this eventually will produce carbon dioxide from the oxidation of acetyl-CoA. We reduce our electron carrier molecules, so NADH and FADH2. We also form a GTP. Now, the reason I wanted to go ahead and write out this entire overall reaction of the citric acid cycle is because when I was first learning the cycle, I kind of often got stuck in the individual reactions that were taking place in this kind of merry-go-round and getting confused by all these names like isocitrate and succinyl-CoA. And I think that when we're trying to understand in particular how this cycle is regulated, that is, trying to figure out when essentially this cycle is in full speed and when it's kind of slowing down, it's actually nice to kind of step back and look at the big picture. Now, the first point I want to make is that in contrast to something like glycolysis, which we usually think about as a metabolic pathway that we really consider to be either on or off, the citric acid cycle is something that we usually consider always to be on, but to various degrees depending on kind of the energy needs of the cell. And the reason it always has to be on, right, is because it needs to be delivering these high energy electron carriers to the electron transfer chain to allow at least some constant flow of ATP production, which is vital to a lot of the tissues in our body. In contrast, you can probably think of a case when, a theoretical case perhaps, where someone might not have any glucose in their diet. And of course, glycolysis then could be off, but the body might be able to use fatty acids and other kind of sources of kind of fuel to enter the citric acid cycle. And indeed, kind of a clinical proof of this is that, there are kind of disease states that involve mutations of enzymes in glycolysis, for example, but you would be hard-pressed to find a living individual who has a mutation in their citric acid cycle because this cycle is so vital to life. So that brings me to my first point, which is that there is no hormonal control in the citric acid cycle because it's on regardless of whether we're in the fed state or the fast state. Instead, the major form of regulation of the citric acid cycle is through allosteric regulation. So I'll remind you that allosteric regulation is simply the ability of a molecule floating around in a cell to bind to a specific enzyme that's part of this pathway, and it binds to a part of the enzyme that's not the active site. And essentially, by doing this, it causes the enzyme to undergo a conformational change, and it can either make the enzyme work better, in which case we call it an allosteric activator, or it can make the enzyme not work as good, in which case we call it an allosteric inhibitor. And the way that I remember that allosteric regulation is a major kind of form of regulation in the citric acid cycle is I remind myself that since the cycle is always on, it still kind of wants to be able to adjust on a minute-to-minute, so rather fast way, to the kind of energy needs of the cell. So I'm going to go ahead and write that here. It wants to respond to the energy needs of the cell. And the way that it can figure this out is by looking at the molecules it has floating around. And these molecules are often those that are involved in regulating this pathway allosterically. And finally, a third way that this cycle is regulated is by looking at substrate availability. And this is exactly what it sounds like. Essentially, if the body doesn't have a lot of acetyl-CoA around, for example, remember, this is one of the major kind of substrates for this pathway, then it makes sense, of course, that the speed by which this NADH and FADH2 is produced is going to slow down because there's just not sounds like. Essentially, if the body doesn't have a lot of acetyl-CoA around, for example, remember, this is one of the major kind of substrates for this pathway, then it makes sense, of course, that the speed by which this NADH and FADH2 is produced is going to slow down because there's just not enough to enter the cycle. Now, one high-yield example of this is when citrate, under conditions of high ATP, generally shuttles a lot of its acetyl-CoA into the cytoplasm for fatty acid synthesis. And of course, when this happens, it's taking the citrate out of the cycle, and so it will slow down the overall cycle. On the flip side, let's say our body was in a very starving state. We haven't had food for quite some while, and in some cases, amino acids can actually begin to break down from our muscles and enter in various places along the citric acid cycle. And one place they enter is they actually are converted into alpha-ketoglutarate here. And suffice to say that the general idea here is that if you have more of this substrate around, it's going to push the cycle to go faster, which makes sense in this case, right? Because the body is alerted to the fact that it's starving, and so it wants to be able to produce more NADH and FADH2 to produce more ATP. All right, so that's kind of a general overview of substrate availability, but now I want to talk more in depth about this allosteric regulation. Now, you wouldn't know this if I told you, but it turns out that there are 3 reactions in the citric acid cycle that have a very large negative delta G. And remember, a large negative delta G means that these reactions are largely irreversible, which means that they are good targets for regulation because once these reactions essentially occur, it's usually like a ball rolling down a hill and will allow everything else to occur. And these 3 reactions are the conversion of oxaloacetate and acetyl-CoA into citrate, as well as the conversion from isocitrate to alpha-ketoglutarate and alpha-ketoglutarate to succinyl-CoA. Now, to go ahead and keep this kind of diagram as clear as possible, I'm going to go ahead and kind of abbreviate the names of these enzymes, but of course you can always go to Wikipedia or a textbook and remind yourself what these enzymes are called. But of course, this enzyme from oxaloacetate and acetyl-CoA to citrate is citrate synthase, and isocitrate to alpha-ketoglutarate is isocitrate dehydrogenase, so I'm going to say ID, and then alpha-ketoglutarate to succinyl-CoA is alpha-ketoglutarate dehydrogenase. Now, the allosteric regulators of these 3 enzymes can end up being kind of a long list, but my hope in this video is to just be a resource for you to come back to and to kind of justify why certain things are allosterically inhibiting or activating these enzymes. So why don't we start off with the allosteric inhibitors? And one kind of easy allosteric inhibitor to remember is NADH because it allosterically inhibits all 3 of these enzymes. And the reason it does this, and it should be apparent to you why this is so, is that notice that NADH is a product of the overall citric acid cycle, right? So it's a product right here. And so if we're building up NADH, it's essentially a sign to the body that the citric acid cycle is going faster than essentially the electron transport chain, which is using up these NADH, can consume those NADHs. And so it's time for the citric acid cycle to slow down. Now, another allosteric inhibitor that should make some sense to you is ATP. So if we have a lot of ATP in the body, it makes sense that this would be an allosteric inhibitor of processes that produce energy, right? Because we want to conserve energy and not kind of make more energy than our body needs. So in this case, it turns out experimentally, for some reason, only 2 of these enzymes have been shown to be inhibited by ATP, and those are citrate synthase and isocitrate dehydrogenase. Now, the final allosteric inhibitors to talk about are actually products that form in the citric acid cycle, and these products, if they accumulate in excess amounts, can actually negatively feed back by allosterically inhibiting some of these enzymes. And the 2 notable products that do that are first citrate, which can allosterically negative feedback on its enzyme citrate synthase, as well as succinyl-CoA, which not only negatively allosterically feedbacks onto the alpha-ketoglutarate dehydrogenase, but it also can actually negatively feedback on the citrate synthase enzyme as well. And one way that I kind of remember why succinyl-CoA might want to kind of feedback all the way back to the citrate synthase is to recognize that the citrate synthase is the kind of first kind of point of entry into the citric acid cycle. And so, if it can stop the citric acid cycle sooner, it will essentially waste less energy, so to say. All right, so that kind of sums up the allosteric inhibitors. But what about the allosteric activators? The first allosteric activator that always comes to my mind is ADP. Remember that ATP is hydrolyzed by a water molecule into ADP and a free phosphate group. So if ATP levels accumulate in excess of ATP, then it's basically a sign that the cell is running out of its power. It's running out of its ATP, and it will therefore need to produce more ATP. And so if it needs to produce more ATP, it makes sense that it would want to activate the enzymes in the citric acid cycle. And it's easy to remember because it activates the same enzymes that ATP inhibits. So those are citrate synthase and isocitrate dehydrogenase. All right, so we're hitting the home stretch here, and there's actually only one more allosteric activator that we need to talk about, and that is calcium. So why might calcium be an allosteric activator? Well, remember that our muscle cells require an influx of calcium to contract. So presumably, if we're exercising really hard, of course, our energy needs go up, but our calcium levels inside of our cell are also going up because of all that muscle contraction. So this is essentially a way for the body, especially in skeletal muscles, to essentially couple muscle contraction with producing more ATP to meet the needs of those contracting muscles. And again, this is experimental evidence that calcium has been shown to allosterically activate isocitrate dehydrogenase, as well as alpha-ketoglutarate dehydrogenase. And again, this is experimental evidence that calcium has been shown to allosterically activate isocitrate dehydrogenase, as well as alpha-ketoglutarate dehydrogenase. Activate isocitrate dehydrogenase, as well as alpha-ketoglutarate dehydrogenase.
After being done with glycolysis in the Krebs cycle, we're left with 10 NADHs, and 2 FADH2s. And I told you that these are going to be used in the electron transport chain, and they're all sitting in the matrix of our mitochondria. And I said they're going to be used in the electron transport chain in order to actually generate ATP. So that's what I'm going to focus on in this video. The electron transport chain. And just so you know, a lot of this stuff is known, but some of the details are actually current areas of research. People have models and they're trying to substantiate the models, but things are happening at such a small scale here that people can just look at the evidence, some of it of which is indirect, and say this is probably what's happening. Most of this is very well established, but some of the exact mechanisms of, for example, how exactly some of the proteins work aren't completely understood. So I think it's very important for you to understand that this is kind of at the cutting edge. You're already there. So that the basic idea here is that the NADHs, and that's where I'll focus, FADH2 is kind of the same idea, although its electrons are at a slightly lower energy state, so they won't produce quite as many ATPs. Each NADH, maybe I'll write it here, each NADH is going to be, as you'll see, indirectly responsible for the production of 3 ATPs, and each FADH2 in a very efficient cell, in both of these cases, will be indirectly responsible for the production of 2 ATPs. And the reason why this guy produces fewer ATPs is because the electrons that he has going into the electron transport chain are at a slightly lower energy level than the ones from NADH. So in general, I just said indirectly, how does this whole business work? Well, in general, NADH, when it gets oxidized, NADH, remember, oxidation is the losing of electrons or the losing of hydrogens that happen to have electrons. We can write its half reaction like this, its oxidation reaction like this. You'll have some NAD+, which you can then go and use back in the Krebs cycle and in glycolysis. You have some NAD+, you'll have a proton, right? A positive hydrogen ion is just a proton. And then you'll have 2 electrons. This is the oxidation of NADH. Oxidation of NADH. It's losing these 2 electrons. Oxidation is losing electrons. Oil rig. Oxidation is losing electrons, or you can imagine it losing hydrogens from which it can hog electrons. Either one of those is the case. Now, this is really the first step of the electron transport chain. These electrons are transported out of the NADH. Now, the last step of the electron transport chain is you have 2 electrons. And you could view it as the same 2 electrons if you like. 2 electrons plus 2 hydrogen protons. And obviously, if you just add these 2 together, you're just going to have 2 hydrogen atoms, which is just a proton and electron, plus 1 oxygen atom. So I can say 1/2 of molecular oxygen.
You have a what?
Plus 2 electrons. And you could view it as the same 2 electrons if you like. 2 electrons plus 2 hydrogen protons. And obviously, if you just add these 2 together, you're just going to have 2 hydrogen atoms, which is just a proton and electron.
Yeah, because with this you'd have a hydrogen anion, right? Which is what's the name of that? Hydrogen anion. Hydride.
Plus 1 oxygen atom, so I can say 1/2 of molecular oxygen. That's the same thing as saying 1 oxygen atom. And you're going to produce, if I have 1 oxygen and 2 complete hydrogens, I'm left with water. And you could view this, we're adding electrons or we're gaining electrons to oxygen. Oil rig, reduction is gaining electrons.
That up pretty
so this
bad.
is the reduction of oxygen to water. This is the oxidation of NADH to NAD+. Now, these electrons that are popping out of, these electrons right here, that are popping out of this NADH, and when they're in NADH, they're in a very high energy state. And what happens over the course of the electron transport chain is that these electrons get electron plus 1 oxygen atom, so I can say 1/2 of molecular oxygen. That's the same thing as saying 1 oxygen atom. Complete hydrogens, I'm left with water. And you could view this, we're adding electrons or we're gaining electrons to oxygen. Oil rig, reduction is gaining electrons. So this is the reduction of oxygen to water. This is the oxidation of NADH to NAD+. Now, these electrons that are popping out of, these electrons right here, that are popping out of this NADH, and when they're in NADH, they're in a very high energy state. And what happens over the course of the electron transport chain is that these electrons get transported to a series of, I guess you could call them transition molecules, but these transition molecules, as the electrons go from one to the other, they go into slightly lower energy states. And I won't even go into the details of these molecules with coenzyme Q, and cytochrome C, and then they eventually end up right here, and they're used to reduce your oxygen into water. Now, every time an electron goes from a higher energy state to a lower energy state, and that's what it's doing over the course of this electron transport chain, it's releasing energy. So energy is released when you go from a higher state to a lower state. When these electrons were in NADH, they were in a higher state than they are when they bond to coenzyme Q. So they release energy. Then they go to cytochrome C, release energy. Now that energy is used to pump protons across the cristae, across the inner membrane of the mitochondria. And this is all very complicated sounding. And this is the cutting edge. So it maybe should sound a little complicated. So let me draw a mitochondria. So let me draw small mitochondria just so you know where we're operating. That's its outer membrane. And then its inner membrane, or its crista, will look like that. Maybe it looks just like that. And let me zoom in on the membrane. So let's say if I were to zoom in right there. So if I were to zoom that out, that box will look like this. You have your crista here, and I'm going to draw it thick. Right, so I'm zooming in. This green line right here, I'm going to draw it really thick. Color it in with green, just like that. And then you have your outer membrane. This outer membrane, I can move it up here. Just color it in. You don't have to see the outside of the outer membrane. Right here, this space right here, this is the outer compartment. And then we learned in the last video This base right here is the matrix. This is the matrix. This is where our Krebs cycle occurred and where a lot of our NADH, or really all of our NADH, is sitting. So what happens is every time NADH gets oxidized to NAD+, or each of and the electrons keep transferring from one molecule to another, it's occurring in these big protein complexes. I'm not going to go into the details of this. So each of these protein complexes span. So let's say that's the protein complex where this first oxidation reaction is occurring and releasing energy. And then let's say there's another protein complex here where the second oxidation reaction is occurring and releasing energy. And these proteins are able to use that energy to essentially pump, this might all seem very complicated, to essentially pump hydrogens into the outer membrane. It actually pumps hydrogen protons, let me be very clear, hydrogen protons into the outer membrane. And every one of these reactions pump out a certain number of hydrogen protons. So by the end of the electron transport chain, or we just followed one set of electrons, by the time that they've gone from their high energy state in NADH to their lower energy state in water, by the time they've done that, they've supplied the energy to these protein complexes that span our cristae to pump hydrogen from the matrix into the outer membrane. So really the only byproduct of the oxidation of NADH into eventually water, or the oxidation of NADH and the reduction of oxygen into water, isn't ATPs yet. It's just this gradient where we have a lot higher hydrogen proton concentration in the outer compartment than we do in the matrix. Or you could say that the outer compartment becomes a lot more acidic. Remember, acidity is just hydrogen proton concentration, the concentration of hydrogen protons. So the byproduct of this, all of this energy is used to really just pump these protons into the outer membrane. So you have two things. The outer membrane becomes more acidic than the matrix inside. Maybe we can call that basic. And then obviously these are all positively charged particles. So there's actually an electric gradient, electric potential between the outer membrane and the inner membrane. This becomes slightly negative, that becomes slightly positive. These guys wouldn't naturally do this on their own. If this is already acidic and it's already positive, left to its own devices, these more protons wouldn't be entering. And the energy to do that is supplied by electrons going from high energy state in NADH to going to a lower energy state eventually on the oxygen in the water. That's what's happening. But essentially all that's happening is protons being pumped from the matrix into the outer compartment. Now, once that gradient forms, these guys want to get back in. These guys want to get back in to the matrix, and that is where the ATPs are formed. So there's a protein that also spans this. Let me draw. So remember, this is all this inner membrane right here. Let me just draw it a little bit bigger right here. So that's our inner membrane, our cristae right there. There's a special protein called, and I'll show you actually a better diagram of what it looks like in a second, called ATP synthase. ATP synthase. And what happens is, remember, because of the electron transport chain, we have all of these hydrogen ions up here, all of these protons really, all they are is protons that really want to get back into the matrix down here. They really want to get back down to the matrix, but they can't. This cristae is impermeable to them, so they have to find a special way to get through. They were able to go the reverse direction through these special protein complexes. Now they're going to go back into the matrix through ATP synthase. So they're going to go back, but something interesting happens. And this is really an area of current research where people are, they think they know how it works, but they're not sure, because you can't just take these proteins apart and operate them or just watch them operate like you can a regular mechanical engine. These are ultra small, and they have to be in a living system, and they have to have the right conditions, and you can't even, it's hard to see hydrogen protons. These are ultra small things that are pretty much, you can't see them. But what happens, the current model is as these enter, as these go through my ATP synthase, there's actually an axle. So you can kind of view this as a housing, and then there's an axle. And this is all just a big protein. There's an axle, and then there's another part of the synthase down here. So you can imagine, this is kind of mind-blowing, that such something this fancy is occurring on the membranes of pretty much all living systems cells. It's not just eukaryotes, even prokaryotes do this. They don't do it in mitochondria, they do it in their cellular membrane, but it's a pretty neat thing. And what happens is, as these go through, you can kind of imagine as water flowing through a turbine, it mechanically causes these kinds of pretty much all living systems cells. It's not just eukaryotes, even prokaryotes do this. They don't do it in mitochondria, they do it in their cellular membrane, but it's a pretty neat thing. And what happens is, as these go through, you can kind of imagine as water flowing through a turbine, it mechanically causes this structure in the middle to spin, to actually spin. This is the current thinking. And this thing is all uneven. It's not like this nice tube, it'll look all crazy like that. And what happens is that an ADP molecule, let's say that this is the A part of the ADP, and then you have two phosphate groups. two phosphate groups, it'll attach to one part of this protein, and maybe a phosphate will just randomly attach to another part of this protein, just like that. So right now it's just ADP and a phosphate. But as this inner axle turns, because it's not a symmetrical tube, it has different things sticking out that has different amounts of atomic charge. It's going to play with this outer housing right here. And so as this turns, the outer housing, because of just the proteins bumping against each other and electrical charge and whatever else, it's going to squeeze the ADP and the phosphate together to actually form ATP. And actually the current thinking is it does it on 3 different sites simultaneously. So as it spins around, ADP and phosphate groups kind of show up on the on the inside of this housing. You can imagine like that. And I don't even know if it's on the inside, but they show up on the housing. And then as this thing spins around, it stretches and pulls on this outer part and pushes these two things together. So it's using the energy from this proton gradient to drive this axle. And because it's all strange, it does all these distortions on this outer part and actually pushes the two ATPs together. So when you start off with your 10 NADHs, it'll provide just enough energy to do just enough protons to put into the outer membrane that when they go back through our ATP synthase, you can almost do our ATP synthase motor, just based on people's observations, they see that this will produce on a per NADH level, roughly 3 ATPs. On a per FADH2 level, roughly 2 ATPs. And I've said multiple times in videos, and this is kind of an ideal. But a lot of times, maybe you'll have some protons leak, so their energy can't be captured properly. Or maybe some of these electrons might somehow jump the gun or jump some steps, so some of the energy gets lost. So you don't always have a completely efficient system. And just so you believe that this is actually occurring on our membrane, there's actual visual depictions of these proteins. This is the actual protein structure of ATP synthase right here. That is actually ATP synthase.
It's pretty cool. Stator.
And as you can see, there's this piece right here that holds this part that holds
Stator. Oh. Great.
that part and that part, you can kind of imagine it relatively stationary. The hydrogen comes through here, the axle gets spun, and as the axle gets spun, ADP and phosphate groups that are lodged inside this F1 part of the protein get pushed together. You have to put energy into the reaction in order to make them stick together, but they get pushed together by the protein itself as this axle turns around. And this axle turns around from the energy of the hydrogen going. I don't even know what the mechanics would look like, but you can imagine, in my head, I imagine kind of, the simplest thing is a windmill, or not a windmill, is maybe some type of water turbine, or maybe the simplest thing is, if you have something like that, I don't know if that's what protein actually looks like. You have any kind of thing passing by, it's going to spin it. It's going to spin it like that. And you could be more creative if you want to change the angle of spin and whatnot. And that's all, people are really still trying to understand this at a deeper and deeper level. But for your purposes, especially at an introductory biology level, you just have to realize that two things are happening in the electron transport chain. Electrons are moving from the NADHs and the FADH2s to eventually show up and reduce the oxygen. And as they do that, they're releasing energy as they go from one molecule to another. They're going to lower energy states. That energy is used to pump hydrogen protons into the outer compartment of the mitochondria. And then that gradient, those hydrogen protons want to get back into the matrix of the mitochondria. So as they go back in, that drives this ATP synthase engine, which actually produces the ATP. So just like we said in the past, when you have 10 of these, on average, let me say it this way, on average, each NADH is going to produce 3 ATPs. Not directly. It produces enough of a gradient of hydrogen protons to produce 3 ATPs in the ATP synthase, and each FADH2, on average, produces enough of a hydrogen gradient to produce 2 ATPs. So if we come in with 10 NADH, they're going to produce, in this ideal world, 30 ATP. And then our 2 FADHs, H2s, are going to produce 4 ATP. And then if you remember, from glycolysis, we had 2 net ATPs directly produced, and from the Krebs cycle, we had 2 ATPs directly produced. So then you have 4 from glycolysis and Krebs, and that gets us once again to our magic 38 ATP from one molecule of glucose. And now I think you have a pretty good grasp of cellular respiration.
585. What the heck? All right, let's take a 5-minute. I should just take a 10-minute break. All right, I'll be back. Yep. Actually. I could do it here, I guess, or something. All right, I'm back. I'll be right back. Hold on. Let me get something real quick. All right, I'm back. Harris Johnson. MCAT score? I don't know yet, buddy. I haven't taken it. I know I'm not ready right now, so I'm not going to waste my time. I'll take it after I feel ready. I know I'm not ready right now. Let's wake up a little bit, eh? Oh. Oh, it's cold. Feels good though. Maybe give me like 2 weeks or something, okay, buddy? I think biology was one of the more dense subjects with a lot of things. I think biochemistry is kind of short. And the rest are kind of short too. PS is just memorization. Well, physics is there's a lot in physics. Physics has a lot of things too. Okay, let's read it up. I'll read it down. Mm. Oh, he didn't get it. He didn't get an answer. I want to know. I want to know. I want to know. Oh, that makes sense. Wow. Accumulating complex 4. Walking the chain. Yeah, that'd be pretty bad. Gotcha. Cool.
The electron transport chain video, and I just wanted to correct it in this one. And it's also an opportunity for me to include a little bit of
So I got more energy now.
Terminology that I forgot to include in that video. So when I described the electron transport chain, we remember it's just you have some high energy electrons in NADH, and they get transferred from one molecule to another, and as they get transferred, they go into lower energy states, and they release energy. And then the final electron acceptor was oxygen. Oxygen got reduced right here. But if you look at both sides of this equation, the mistake was I need two hydrogens. If I have two hydrogens on the right-hand side of the water, I need two hydrogens on the left-hand side. So there should be there should be a 2 right there. So that was what I would consider to be a minor mistake in the last video. But this also gives me a chance to introduce you to some more terminology. So this whole process, we know that this is called oxidation, right? When NADH loses a hydrogen, remember oxidation is losing formally electrons. When it loses the hydrogen, it loses the opportunity to hog that hydrogen's electrons. So this whole process of the electron transport chain is one molecule after another getting oxidized until you have a final electron acceptor in water. So this is, obviously you could call this oxidation, just very generally. And then the second part of the electron transport chain, or maybe we shouldn't even call this part of the electron transport chain, the process where the ATP is actually formed, the adding of a phosphate group to another molecule is called phosphorylation. Phosphorylation. So the whole process of creating ATP through the electron transport chain. Remember, the electron transport chain releases energy that creates this hydrogen gradient. It pumps the hydrogens into the outer compartment, and then that gradient, those hydrogens that want to get back into the matrix, essentially go back through this ATP synthase. This process of generating ATP this way is called oxidative phosphorylation. Oxidative phosphorylation. It's a good word to know. You might see it on some standardized tests or on your exams. And it's called this because you have an oxidative part. Each of these molecules get oxidized in the electron transport chain as they lose their hydrogens or as they lose their electrons. That creates a hydrogen gradient. And then that, through chemiosmosis, allows for phosphorylation. So that's another good word to know. The transfer of these hydrogens, these kind of going through this membrane selectively. This membrane, ATP synthase won't allow just any molecule to go through it. It's allowing these hydrogen protons to go through it. This process right here of this hydrogen going through it is called chemiosmosis. Chemiosmosis, another good word to know. So the entire process called oxidative phosphorylation, they don't happen at the same time. Oxidative generates the energy because the energy to push the hydrogens out, and the phosphorylation happens as the hydrogens experience chemiosmosis and go back in and turn this little axle and then push the ATP or the ADP and the phosphate groups together. And then you can contrast that with substrate phosphorylation, since I'm in the mood to introduce you to terminology. Substrate phosphorylation. This is actually what happens when the ATP is produced directly in glycolysis and the Krebs cycle. So this is in glycolysis and the Krebs cycle. And this is where you have an enzyme directly helping to produce the ATP without any type of chemiosmosis or proton gradient. So if you imagine an enzyme, some blurb, some big protein blurb, and let's say it has the ADP there with its two phosphate groups, and then maybe it has another phosphate group that attaches to some other part of the enzyme. This enzyme facilitates without any kind of chemiosmosis or oxidation. It facilitates probably in conjunction with other energy releasing reactions that maybe are occurring on other parts of the enzyme. So maybe you can imagine a little spark right there, and then that twists this entire enzyme. This is exactly how it might work, but without any type of chemiosmosis or proton. Oxidative generates the energy because the energy to push the hydrogens out, and the phosphorylation happens as the hydrogens experience chemiosmosis and go back in and turn this little axle and then push the ATP or the ADP and the phosphate groups together. And then you can contrast that with substrate phosphorylation, since I'm in the mood to introduce you to terminology. Substrate phosphorylation. This is actually what happens when the ATP is produced directly in glycolysis and the Krebs cycle. So this is in glycolysis and the Krebs cycle. And this is where you have an enzyme directly helping to produce the ATP without any type of chemiosmosis or proton gradient. So if you imagine an enzyme, some blurb, some big protein blurb, and let's say it has the ADP there with its two phosphate groups, and then maybe it has another phosphate group that attaches to some other part of the enzyme. This enzyme facilitates without any kind of chemiosmosis or oxidation. It facilitates probably in conjunction with other energy that maybe are occurring on other parts of the enzyme. So maybe you can imagine a little spark right there, and then that twists this entire enzyme. This is exactly how it might work, but it's a good idea. And then these two things maybe get pushed together. When it's just an enzyme without any of this chemiosmosis that's driven by oxidation, like we learned in the electron transport chain, we call this substrate phosphorylation. And the substrates are just the things that attach to the enzyme and have something performed on them. So anyway, hopefully you found this little video mildly useful.
In talking about how oxidative phosphorylation is actually regulated inside of our cells, I find it helpful to remind myself of two kind of basic things about this pathway. So the first is, what is the purpose of oxidative phosphorylation? So remember, this is a process that takes place inside of the mitochondria in the electron transport chain, right? And its sole purpose is to produce lots of ATP. So remember, ATP can be produced without oxygen through substrate level phosphorylation, which does take place in glycolysis and also the Krebs cycle. But what's cool about electron transport chain and oxidative phosphorylation is that by having oxygen and having the electrons shuttled through these electron carrier molecules like NADH and FADH, it allows the body to produce efficiently a whole lot of ATP, which is important for many of our tissues which can't survive just on substrate level phosphorylation. So for example, your brain and your heart and some other tissues in your body really rely on the electron transport chain to produce most of its ATP. And the second point that's important to recognize is that oxidative phosphorylation is the kind of common end pathway of aerobic respiration. So what do I mean by this? So remember that there are many different types of fuels that can enter cellular respiration. So we've talked about glucose and also fatty acids can enter cellular respiration, as well as occasionally, under extreme starvation, amino acids can also enter. But ultimately, all of these are broken down, and much of their reducing power is stored in the electron carrier molecules like NADH and FADH2 that are ultimately shuttled, as I mentioned before, to the electron transport chain to produce this ATP. All right, so how do these 2 points relate back to how oxidative phosphorylation is regulated? Well, this first point simply reminds me that the major form of regulation in oxidative phosphorylation is looking at the energy needs of the cells. And the way that the body does this is by looking at the levels of ADP compared to the levels of ATP. And specifically, it should make sense to you that if the body has a lot of ATP lying around, it should essentially be assigned to say, you know what, we have enough energy, we don't need to produce more. Oxidative phosphorylation can slow down. But on the other hand, if we have a lot of ADP compared to ATP, it's a sign that the cell is running out of ATP and that more ADP can be and should be phosphorylated using the electron transport chain. And this is actually just really a kind of application of Le Chatelier's principle, which is a general chemistry principle to oxidative phosphorylation. And I'll actually go into a little bit more detail about this in a second. But first, I want to kind of touch on the second point here, which is that it's a common end pathway for aerobic respiration. And I really make this point because it reminds me why there is kind of no major hormonal or allosteric. Remember, allosteric means that there's some type of enzymatic control that's being altered, but there is no major hormonal or allosteric regulation in oxidative phosphorylation. And the way I kind of always justified that to myself is that these are these forms of regulation allow us to really fine-tune regulation and to make sure that when we turn something on, we are turning it on with full certainty. But the fact that it's downstream of many of the entry points to aerobic respiration, such as breaking down glucose and glycolysis and the oxidation of fatty acids, means that it's probably more important for those pathways, which they in fact do have a lot of hormonal and allosteric regulation. But once those pathways are turned on, it's kind of just going to keep rolling down the pathway. And it probably may not be as important to have that level of kind of fine tuning in oxidative phosphorylation. So with that in mind, let's go ahead and talk more about how the energy levels in the body are used to regulate oxidative phosphorylation. So I've gone ahead and drawn out a simplified diagram of the electron transport chain. And I want to remind you that it's much more complex than this, right? We have 4 protein complexes, we have ATP synthase, we have this all occurring in the inner mitochondrial membrane. But for our purposes, I just really wanted to highlight what the main reactants and products of the electron transport chain were. So let me go ahead and guide you through this. So we have the entry of electron carrier molecules such as NADH. And remember, we can also be dealing with FADH2 as well, but just, as an example, I'm using NADH. And the NADH carries 2 electrons from the molecule, from the fuel, such as glucose, or it could be fatty acid or some type of fuel. And it essentially gets oxidized at the electron transport chain. It releases its electrons into the electron transport chain and becomes itself oxidized. This flow of electrons, of course, fuels the phosphorylation of ADP and a free phosphate group into ATP. Of course, this is all done indirectly through a proton gradient that's formed in the intermitochondrial membrane. electrons must have somewhere to go, and they end up reducing oxygen. It's kind of funky to think about 2 electrons reducing half an oxygen, but this is just so that the stoichiometry works out. You can see here that if we were to reduce 1 molecule of oxygen, of course, it reduces oxygen and combines with some electron transfer chains are a chemical reaction that's in equilibrium, and there is some type of alteration to this equilibrium. So let's say we have the addition of more reactant, or we take away. Le Chatelier's principle essentially says that the equilibrium will re-equilibrate to counter this change. So it turns out that this is exactly how the electron transfer chain is regulated. And to make this point, let's go ahead and basically ask ourselves what would happen if we had more NADH, more ATP, or free phosphate, or more oxygen around. So remember, these are all of our reactants. And indeed, if we had more of these reactants, the Chatelier's principle would essentially say that this reaction, so to say, would be pushed towards the forward direction, and we would produce more ATP. So we would say that that kind of flow of electrons through the electron chain is faster, and we get more ATP. Now, of all 3 of these reactants, I just want to make a point here that practically speaking, we consider the level of oxygen to be pretty constant. If we're breathing in and out normally this is not a limiting factor that essentially alerts the electron transport chain to go faster. So I'm just going to go ahead and erase that for practical purposes. But generally speaking, of these 3, the NADH, the ATP, and the free phosphate group, it's really the levels of ADP in the cell that are most likely to alert the electron transport chain to produce more ATP. And that's just because it's usually the limiting factor of all 3. But it should make sense to you that high levels of NADH are essentially a sign from up above, from the breakdown of glucose or fatty acids, that it's time to make more energy for the cell. Of course, we can also do this thought experiment for the products of the reaction. So specifically, let's say we had elevated levels of ATP in the cell, or elevated levels of the oxidized form of these electron carrier molecules. Le Chatelier's principle would tell us that this equilibrium would essentially shift in the opposite direction. So the flow of electrons through would be slower, and we would produce less ATP. Of course, in reality, we don't really think about electrons traveling the opposite direction down the electron transport chain, but this is just a way to kind of essentially signify that having higher levels of ATP in the body, or high levels of NAD+, are essentially, by Le Chatelier's principle, putting a brake on the electron transport chain. And just as before, the ADP levels from all 3 of these reactants makes sense to you that high levels, specifically, let's say we had elevated levels of ATP in the cell, or elevated levels of the oxidized form of these electron carrier molecules. Le Chatelier's principle would tell us that this equilibrium would essentially shift in the opposite direction. So the flow of electrons through would be slower, and we would produce less ATP. Of course, in reality, we don't really think about electrons traveling the opposite direction down the electron transport chain, but this is just a way to kind of essentially signify that having higher levels of ATP in the body, or high levels of NAD+, are essentially, by Le Chatelier's principle, putting a brake on the electron transport chain. And just as before, the ADP levels were more likely to alert the electron transfer chain, ATP levels are kind of the limiting factor to alert the electron transfer chain as compared to, the NAD+ levels. And that's really because the body usually keeps NAD+ and NADH in a pretty kind of stable ratio. So the body's really looking to whether there's high levels of ADP or ATP to ultimately decide and regulate how fast the electron transfer chain is.
Thank you, ma'am.
All right, so if we were going to go on the ambitious task of tallying up how much ATP was produced in one cycle of cellular respiration, or just to be super clear here, I mean how much ATP was produced per the oxidation or breakdown of 1 molecule of glucose in cellular respiration, we might start off by just getting ourselves organized and reminding ourselves that there are 2 kind of main ways that we produce ATP in cellular respiration. So the first minor contribution comes from something called substrate level phosphorylation. And remember that this is exactly what it sounds like. We have a substrate or a molecule, I'm just going to say R. And remember that in the context of cellular respiration, this is usually, we think of this as a kind of metabolite, an intermediate metabolite of glucose. So somewhere along glucose's oxidation, we get a metabolite, and we activate this metabolite with a phosphate group. And from this phosphate group, we can actually donate it directly to ADP to produce ATP. And of course, our molecule also gets modified in the process, usually gaining a hydroxyl group. But the details aren't entirely important, except to realize that this phosphorylation is occurring at the level of a substrate. This is in contrast, of course, to oxidative phosphorylation, which is where we get the bulk of our ATP. And this oxidative refers to the fact that this process requires oxygen. And in fact, the importance of oxygen here is that this oxygen is reduced by electron carrier molecules in something called the electron transport chain. So remember that we have electron carrier molecules called NADH and FADH2 that are produced at various stages of cellular respiration, glycolysis, the oxidation of pyruvate, the Krebs cycle, and it's basically storing up all of that energy from the glucose molecule, and it's going to donate it into the electron transfer chain. And of course, the final electron acceptor is oxygen, which is then reduced to water. But the important part here is that this flow of electrons is able to power something, essentially fuel something called ATP synthase, which is an enzyme that is in the mitochondrial membrane that produces the bulk of our ATP. Now, the next point I want to make here is that it's actually been possible for us to calculate the exact number of ATP produced in substrate level phosphorylation, and we've also nailed down the amount of NADH and FADH2 molecules that are produced in this process as well. But for quite a while, it was difficult to nail down the exact number of ATP molecules that were produced in oxidative phosphorylation. And for this reason, actually, and I'll get back to kind of why we're unable to kind of nail down a number here. But for this reason, you might often see quite a range of predictions for how much ATP is actually produced in one cycle of cellular respiration. Just to give you an idea of that, when I look at some textbooks, you can see a range of anywhere from 30 to 38 molecules of ATP that are predicted to be produced for the oxidation of one molecule of glucose. So of course, to get back to this kind of elusive calculation of ATP, researchers have done controlled studies in which they basically take a known amount of NADH or FADH2, and they have mitochondria available in the lab, and they basically allow the mitochondria to oxidatively phosphorylate these molecules and essentially measure how much ATP is produced. But kind of to their surprise at first, they found that for NADH, for one molecule of NADH they calculated, there was not a whole number of ATP produced. In fact, they found that there was somewhere between 2 to 3 ATP molecules produced for every one NADH molecule. And for FADH, they also found that there was no whole integer number of ATP, but rather there was a range, somewhere between 1 to 2 ATP produced. Now, for the longest time, researchers kind of looked at these results and said, whole numbers are a lot easier to deal with, and so why don't we just assume, for the sake of assumption, that we can kind of round up and we'll say that for every one molecule of NADH, let's say that we have 3 molecules of ATP produced, and for every molecule of FADH2, we have 2 molecules of ATP produced. And so using these kind of estimations, they calculated essentially the upper range of ATP. So these calculations were ultimately used to calculate kind of this number of 38 ATP produced in cellular respiration, kind of the upper limit of ATP produced. But of course, we still have this range. And in fact, it's worth kind of pausing to stop and think about for a second if it is surprising that we have this range in the first place. And so to think about this a little bit further, I want to go ahead and kind of just draw out, without getting too detailed, kind of a depiction of what's going on in the electron transport chain. So remember that the electron transport chain is taking place in the mitochondria. The mitochondria has 2 membranes. We have the inner mitochondrial membrane, which I'm going to label here as I, and we have the outer mitochondrial membrane. And along the inner mitochondrial membrane, we have a series of proteins that are known as protein complexes. And, these all have specific names, but just for our purposes, it's important to recognize they're kind of available in the lab, and they basically kind of donate it into the electron transfer chain. And of course, the final electron acceptor is oxygen, which is then reduced to water. But the important part here is that this flow of electrons is able to power something, essentially fuel something called ATP synthase, which is an enzyme that is in the mitochondrial membrane that produces the bulk of our ATP. Now, the next point I want to make here is that it's actually been possible for us to calculate the exact number of ATP produced in substrate level phosphorylation, and we've also nailed down the amount of NADH and FADH2 molecules that are produced in this process as well. But for quite a while, it was difficult to nail down the exact number of ATP molecules that were produced in oxidative phosphorylation. And for this reason, actually, and I'll get back to kind of why we're unable to kind of nail down a number here, but for this reason, you might often see quite a range of predictions for how much ATP is actually produced in one cycle of cellular respiration. Just to give you an idea of that, when I look at some textbooks, you can see a range of anywhere from 30 to 38 molecules of ATP that are predicted to be produced for the oxidation of one molecule of glucose. So of course, to get back to this kind of elusive calculation of ATP, researchers have done controlled studies in which they basically take a known amount of NADH or FADH2, and they have mitochondria available in the lab, and they basically allow the mitochondria to oxidatively phosphorylate these molecules and essentially measure how much ATP is produced. But kind of to their surprise at first, they found that for NADH, for one molecule of NADH they calculated, there was not a whole number of ATP produced. In fact, they found that there was somewhere between 2 to 3 ATP molecules produced for every one NADH molecule. And for FADH, they also found that there was no whole integer number of ATP, but rather there was a range, somewhere between 1 to 2 ATP produced. Now, for the longest time, researchers kind of looked at these results and said, whole numbers are a lot easier to deal with, and so why don't we just assume, for the sake of assumption, that we can kind of round up, and we'll say that for every one molecule of NADH, let's say that we have 3 molecules of ATP produced, and for every molecule of FADH2, we have 2 molecules of ATP produced. And so using these kind of estimations, they calculated essentially the upper range of ATP. So these calculations were ultimately used to calculate kind of this number of 38 ATP produced in cellular respiration, kind of the upper limit of ATP produced. But of course, we still have this range. And in fact, it's worth kind of pausing to stop and think about for a second if it is surprising that we have this range in the first place. And so to think about this a little bit further, I want to go ahead and kind of just draw out, without getting too detailed, kind of a depiction of what's going on in the electron transfer chain. So remember that the electron transfer chain is taking place in the mitochondria. And the mitochondria has 2 membranes. We have the inner mitochondrial membrane, which I'm going to label here as I, and we have the outer mitochondrial membrane. And along the inner mitochondrial membrane, we have a series of proteins that are known as protein complexes. And, these all have specific names, but just for our purposes, it's important to recognize there are kind of just 4 main protein complexes. And in some textbooks, people will actually call ATP synthase, which I'm going to go ahead and draw here in yellow, as complex number 5. So let me go ahead and label these.
Hello?
1 through 5, just so we remember that. So these 4 represent the protein complexes that shuttle electrons, and of course,
Thanks, Mahi. Appreciate it.
5 represents ATP synthase. Now recall that the basic premise here is that these reduced electron carriers donate electrons to the electron transfer chain, and in fact, specifically, NADH donates 2 electrons to protein complex number 1, and FADH2 donates 2 electrons to protein complex number 2. Now, the second important point is that as these electrons are kind of flowing down these proteins, for every 2 electrons that kind of flow by, it's actually been calculated that protein complex number 1 pumps 4 protons into the intermembrane space. Protein complex 3 pumps also 4 protons. And protein complex number 4 pumps 2 protons. And protein complex number 2 doesn't really contribute. Now, with these facts in mind, we can go ahead and actually calculate how many protons are pumped for a molecule of FADH2 and how many protons are pumped for a molecule of NADH. So let's go ahead and just quickly do that here. So because NADH donates at the very first electron complex, it contributes to a total of 4 plus 4 plus 2, or 10 protons are pumped out for every molecule of NADH. On the other hand, FADH2 enters in complex number 2, so it only contributes to the total pumping of 6 protons. And so we can say that there are 6 protons that are pumped for every molecule of FADH2. All right, so here's a good place to pause and kind of get back to our original question, which was, we have this kind of range of ATP molecules produced per molecule of NADH or FADH2, and why is it that, we don't have a whole number integer ratio between the amount of ATP and NADH that we have? And the reason kind of that we might be able to justify this looking at our diagram here is that NADH and FADH2 each contribute to this proton gradient, but really it's not NADH or FADH2 that's directly donating anything to form ATP, because remember that it's this proton gradient that forms this intermembrane space here that essentially fuels this ATP synthase. Remember that protons flow back through this ATP synthase molecule, and in doing so, they essentially power this pump to phosphorylate ADP into ATP. And so of course, maybe the question we should really be asking is, how many protons does it take, or how many protons need to flow through this ATP synthase to phosphorylate one molecule of ADP into ATP? And so I'm actually going to go ahead back to our ratios up here and write out here that if we knew how many protons were necessary to produce one molecule of ATP, we might be able to calculate essentially the ratio of ATP to NADH or FADH2. And it's this calculation that I think researchers are actually still trying to, nail down. And, I'm sure depending on the type of cell and the state of the cells, the efficiency of this process is going to be different and might, change moment to moment. And so maybe, the expectation to have an exact number is not realistic, but researchers are pretty confident with the number right now currently of 4 protons being necessary to produce one molecule of ATP. So I'm going to go ahead and just write that in here. And with this number, we can actually go ahead and calculate the ratio of ATP to NADH. And so simply we have here, for every molecule of NADH, we have 2.5 molecules of ATP. And for every molecule of FADH, we have 6 divided by 4, or 1.5 molecules of ATP that are produced. So remember that even though it's kind of funky that we're talking about kind of 2.5 ATP per molecule of NADH or per molecule of FADH2, really what this is alluding to is the role of this chemiosmotic coupling, or using this proton gradient to fuel ATP synthase. And because we're talking about protons now, we need to factor in that, we end up getting these non-whole number ratios between ATP and NADH or FADH2. But with these ratios in mind, I actually want to go ahead and calculate kind of the sum total of ATP that we produce in cellular respiration. So I've already gone ahead and kind of created a table here. And remember that we're talking about one cycle of cellular respiration. So I'll say total ATP yield, let's say per one molecule of glucose, remember? And so I've already kind of written out here how many ATP and electron carrier molecules are produced in glycolysis and the oxidation of pyruvate and the Krebs and TCA cycle. So now let's go ahead and, using our ratios here, let's go ahead and write out how much ATP we have. So 2 ATP is 2 ATP, and 2 NADH, using our conversion factors, 2 times 2.5, which is going to be 5 ATP. And then we have, again, 5 ATP. And 2 ATP here, formed in citrate level phosphorylation, is 2 ATP. And 6 NADH times 2.5 is going to yield 15. And 2 FADH2 times 1.5 is going to yield 3. And so if we add all of this up, we get 32 ATP. Now, before I call it good, I want to make one more last nitpicky point, which is to realize that glycolysis, remember, takes place in the cytosol. So unlike the oxidation of pyruvate and the Krebs cycle, which take place in the mitochondria, the NADH that's produced in the glycolysis must actually be shuttled somehow into the inner mitochondrial membrane in order to donate its electrons into the electron transport chain. But for some reason, it turns out that the inner mitochondrial membrane is actually not permeable to this molecule NADH. So the body has actually come up with something called shuttle transport systems to shuttle this NADH into the mitochondria. And it turns out that depending on where the NADH is shuttled into the electron transport chain, so if we actually go back to our diagram here, some of the electrons from the NADH produced in glycolysis can be shuttled into the first protein complex, and some of them are actually shuttled
I'm lagging. Why am I lagging? Why are you lagging? No. Freaking computer, man.
Into this
There you
third
go. We're
protein
back.
complex here. And so depending on whether it's shuttled earlier or later on in the electron transfer chain, a different number of protons will be pumped into the proton gradient, remember. And so the conversion factor for the amount of ATP produced is going to be different depending on which shuttle is used. So I just want to make that point and have you be aware of the fact that this number right here, this number here, is actually a range. It can actually range from anywhere to 3 to 5 ATP produced per molecule of NADH. I'm going to go ahead and kind of just adjust this to say that really the range here is 30 to 32 ATP produced per 1 cycle of cellular respiration. And this right here is the generally accepted number for the amount of ATP produced in 1 cycle of cellular respiration.
Thank you. Thank you. Oh man! All right, let's take a. Thanks, Mahi. Appreciate you. Thank you so much. Thank you. Yeah, thank you, Happy. Yeah, thanks everyone. Yeah, thank you. Everyone. Yeah. All right, let's take a 15, and we'll come back and we'll finish this one. And I have the questions. I have the questions that I just didn't do, but I will do them. And these questions here are the ones I want, and for the ones I didn't do, so that's gonna take some time. Some of them have 4. It's gonna take quite a while. But before I start that, well, I'm gonna finish this. I mean this one, right? But before I start that, I want to read the high yield guide, which I will too. I'll read this one. This one. I don't know what you guys probably don't see. This one. I'm going to read this one on the stuff we covered. We covered quite a lot. Yeah, a lot of some stuff. Where's the this one? Sliding filament theory for muscle contraction, muscle system, and find whatever other stuff we went over that is in here that we can go over. And and then I'll answer the questions, and then I'll add the cards to Anki, and then I have just a couple of videos I want to watch from Andrew. We'll read the book, and then we'll do Yusuf. I think that's the strategy I want to do. All right, I'll be back. Hey, Chirpy. Hey, Chirpy. Thank you. Appreciate it, buddy. Much love. Thanks. I will follow my dreams. Appreciate you. Follow my dreams. Follow your dreams. All right, I'll be back. Much love, Chirpy. Peace, brother. Peace. Peace, buddy. Peace and love. All right, I'll be back. I'll be back. particularly bad. I should exercise a little bit. At least exercise a little bit, eh? Thanks, Fomi. I appreciate that. And through you, John. Oh, appreciate it. Yeah, thank you. It's quite early, so I think I just want exercise to get it out of the way. Wasn't feeling like it, so. I don't know what we'll do today, though. Thank you. It's Chris. Hey, Chris. It's Christy. Oh, it's Crystal. Hey, Crystal. Thank you, Crystal. Thank you, you too. All right, let's wash my hands, and then we'll get started again. Let's wrap this up, man. We should have been done with this a while ago. Cool boy. Am I excited? Am I excited? Pumped up. Cool. Okay. Reading mode. Yeah. Let's make those leg muscles switch, eh? My legs are definitely the weakest part, and they're the hardest to grow. I've compared to my biceps, which took me a couple of years to build, and I'm still building, but my legs, I think they're pretty much the same. They've been the same for years. Okay. That's a good one. This one's a little bit lower. Maybe here. Yeah, that's good. Check that out. Twitchy leg, huh? Pretty cool. Yeah, that's pretty cool. Okay.
That we built our discussion of metabolism on is that we extract energy from food. And of course, this comes from the fact that we know that ATP is our body's main source of chemical energy. And the way we can produce this ATP is by breaking down nutrients such as glucose and fatty acids and proteins, which are all found in food. But of course, the question that you might wonder is, well, we don't constantly eat food, right? So how does our body produce a constant flow of ATP even though we only have food very intermittently? And of course, the answer to that question is that our body has evolved to store fuel as well, so that we're not reliant on an immediate influx of nutrients right after a meal. So in the remainder of the video, I actually want to go ahead and compare and contrast the three main types of fuel that our body has evolved to be able to store, and then touch on why one of these fuels is actually a much better storage fuel than the rest of them. So let's start off with glycogen. And glycogen is our body's way of storing carbohydrates. And essentially, it's just a long chain or a polymer of glucose molecules that are all attached to each other. And our body stores this mostly in the liver, but also there's some in our muscles as well. And if we were to tally up how much glycogen, how many grams of glycogen that we have, let's say in an average 70 kilogram male, an average healthy 70 kilogram male, we would calculate there would be approximately 480 grams of glycogen. And just to give you some idea of how much energy we can extract from glycogen, we can extract approximately 4 kilocalories of energy per gram of glycogen. And kilocalories is just a unit of energy, and it's something that you might see on cereal boxes or any type of food really when you look at the nutrition label. And just to give you some perspective, the recommended average intake of energy for humans is somewhere around 2,000 kilocalories per day. And of course, it's a huge ballpark number. The exact number. Even though we only have food very intermittently. And of course, the answer to that question is that our body has evolved to store fuel as well, so that we're not reliant on the immediate influx of nutrients right after a meal. So in the remainder of the video, I actually want to go ahead and compare and contrast the 3 main types of fuel that our body has evolved to be able to store, and then touch on why one of these fuels is actually a much better storage fuel than the rest of them. So let's start off with glycogen. And glycogen is our body's way of storing carbohydrates. And essentially, it's just a long chain or a polymer of glucose molecules that are all attached to each other. And our body stores this mostly in the liver, but also there's some in our muscles as well. And if we were to tally up how much glycogen, how many grams of glycogen that we had, let's say in an average 70 kilogram male, an average healthy 70 kilogram male, we would calculate there would be approximately 480 grams of glycogen. And just to give you some idea of how much energy we can extract from glycogen, we can extract approximately 4 kilocalories of energy per gram of glycogen. And kilocalories is just a unit of energy, and it's something that you might see on cereal boxes or any type of food really when you look at the nutrition label. And just to give you some perspective, the recommended average intake of energy for humans is somewhere around 2,000 kilocalories per day. And of course, it's a huge ballpark number. The exact number might fall above or below this and really depends on how old you are, what your sex is, as well as how active you are during the day. Now, a second type of fuel that our body stores is proteins. And remember that proteins are nothing more than a long chain of amino acids. And most of the protein in our body is in our muscles. And again, in an average 70 kilogram man, if we were to tally up how many grams of proteins, we would get around 6,000 grams. And again, similar to glycogen, we will be able to extract about 4 kilocalories of energy per gram of protein. Now, finally, the third type of fuel that our body stores is in the form of fats. And these fats are actually stored up in specialized tissue in our body called adipose tissue. Now, if we were to tally up how many grams of adipose tissue a 70 kilogram man had, it would actually amount to a much higher amount than both proteins and glycogen combined. In fact, it's somewhere around 12,000 grams of fat in an average 70 kilogram healthy male. And moreover, unlike glycogen and protein, we can actually extract a lot more energy per gram of fat. In fact, that comes out to be somewhere around 9 kilocalories per gram of fat. Now, to put this in perspective, let's kind of do a fun, simple math problem here. Let's assume that this 70 kilogram guy requires an intake of 2,000 kilocalories per day. Now, if this is the case, let's kind of ask ourselves a very theoretical question, because of course it would be unhealthy to starve oneself, but if one didn't have an intake of food, how long would this man be able to survive on each type of fuel? So if we round this to about 500 grams of glycogen times 4 kilocalories per gram, that would actually amount to about 2,000 kilocalories that we could extract from glycogen. And so that would last him about a day, right? Now let's move down to protein. So proteins, we have about 6,000 grams in our body times 4 kilocalories per gram, which amounts to about 24,000 kilocalories divided by 2,000. That would last us about 12 days, right? So we're getting a little bit better. But now let's actually take a look at fats. We have substantially more amount of fats in the body than proteins and glycogen, right? So 12,000 times, let's round this up to 10, since we're just approximating anyways. And so if we multiply 12,000 times 10, that's about 120,000, right? Divided by 2,000, that's about 60 days that one could go, theoretically, of course, and probably may not even make it that far, but theoretically, we could make it 60 days on just our fat storage alone. Now, that's pretty impressive, I think. So now the question I want to answer is, why has fat become the major source of storage fuel in our body? And to do that, let's first actually remind ourselves what the chemical structure of fats are. So I've kind of, to save us some time, drawn out the chemical structure of a triacylglyceride. And I'll actually go ahead and write that out here. So triacylglyceride. And just a point of clarification, I am using the word fat and triacylglyceride interchangeably. And that's because fat is just kind of an everyday term that we use to refer to the type of fat, the triacylglyceride, that we store in our body. And so we're talking about the chemical structure. It probably makes sense in this case to refer to it from its chemical name, which is triacylglyceride. And in fact, its chemical name tells us a lot about its structure. So let's take a look. So this triacyl refers to these three acyl groups, these acyl side chains. So what is an acyl side chain? So, an acyl is just a reference to a type of organic chemistry functional group. So there are some functional groups you might be familiar with, like hydroxyl groups or phosphate groups. And in this case, the acyl group is anything that has a carbon double bond oxygen attached to a long chain of carbons and hydrogens. And actually, I should say the chain doesn't have to be long. It just has to be some type of organic functional group. But in this case, the chain happens to be very, very long. And of course, I kind of just gone ahead and drawn three acyl groups that have come pretty much randomly to my mind, because the idea here is that these chains can vary immensely depending on the type of triacylglyceride in our body, depending on the type of fats that we ingest. And so some of them might have single bonds, which we refer to as them being saturated with hydrogens, and some of them might have double bonds, in which case we refer to these triacylglycerides or these side chains as being unsaturated. So that's kind of just some nomenclature that you might see. And then finally, this glyceride refers to the backbone of this molecule, these kind of three carbons that are hanging out down here. And they also have a oxygen attached to them as well. And of course, they link with these acyl side chains through this ester linkage that I'm kind of highlighting in green here. So that's the big picture of this molecule. Now, the reason why I think it's important to be familiar with the structure of a fat when trying to understand why fat is such a prominent type of storage fuel in our body is because now you can visually see where all of that energy, where all that 9 kilocalories per gram of energy is coming from. Because looking at this molecule, you can see the bulk of it is formed by these long carbon-hydrogen chains. And these carbon-hydrogen chains are referred to as being very high energy because they have a lot of electrons kind of stored up in these bonds. And we know, if we remind ourselves back to kind of our general principle, that if you have a reduced organic molecule like this, like we can see here, we are able to extract energy by oxidizing it in kind of subsequent steps. And this flow of electrons can be harnessed by something like the electron transfer chain to allow us to produce ATP. So that's kind of my first point here, and I'll scroll down here and I'll go ahead and write that, which is that the triacylglyceride is a very energy-rich molecule, has lots of these carbon-hydrogen bonds that can be oxidized to produce ATP. Now, a second reason why these triacylglycerides are such a good form of storage energy is because they're relatively chemically inert. So what I mean by this is that they're unlikely to react with other things in the body. And this is, of course, in contrast to things like glucose and proteins, which are quite polar. They have many polar functional groups, like hydroxyl groups, for example, and they can react with a lot of things in the aqueous environment of the body. But triacylglycerides are remember, they're not soluble in water. And just, remind yourself of, if you've ever made salad dressing, it's so hard to mix the oil and water together, right? And so because this isn't, going to dissolve in water, and it has a lot of these, carbon-hydrogen chains that are considered not to be very reactive, it serves as a great form of storage energy because it won't, randomly react or be wasted in side reactions. A third reason why fats are good for energy storage is because they have no large or prominent functional role inside the body, in contrast to proteins, for example, which are used to make enzymes. And enzymes are of course, of paramount importance in our body. And so we wouldn't want to rely too heavily on proteins because it would kind of be a conflict of interest for our body, right? Because if we use up too many proteins, we wouldn't be able to make enzymes. And that's why fats end up being kind of a good compromise, because we're able to kind of essentially just store up fats for one major purpose alone, which is to produce energy. Of course, as a blanket statement, fats are important in other ways as well, but largely speaking, their main role is to store energy. So I've touched on why triacylglycerides are a good form of energy storage and why proteins might not be such a good form of energy storage. But you might be wondering, what about carbohydrates? What about glycogen? Why didn't our body evolve to make glycogen the major storage fuel? And that brings me to my fourth point, which is that unlike proteins or carbohydrates, triacylglycerides fats are very hydrophobic. And a benefit of being hydrophobic, as we kind of touched on earlier, so not only does that make it more inert chemically because it can't react with a lot of things in the water in the aqueous environment, but it also means that it won't be kind of weighed down by water. So you might be wondering, why is that beneficial? Why is it beneficial not to be weighed down by water? And to answer that question, we can actually just go ahead and do kind of a quick math problem here to kind of make it more inert chemically because it can't react with a lot of things in the water in the aqueous environment, but it also means that it won't be kind of weighed down by water. So you might be wondering, why is that beneficial? Why is it beneficial not to be weighed alone, which is to produce energy? Of course, that's a blanket statement. Fats are important in other ways as well, but largely speaking, their main role is to store energy. So I've touched on why triacylglycerides are a good form of energy storage and why proteins might not be such a good form of energy storage. But you might be wondering, what about carbohydrates? What about glycogen? Why didn't our body evolve to make glycogen the major storage fuel? And that brings me to my fourth point, which is that unlike proteins or carbohydrates, triacylglycerides fats are very hydrophobic. And a benefit of being hydrophobic, as we kind of touched on earlier, so not only does that make it more inert chemically because it can't react with a lot of things in the water in the aqueous environment, but it also means that it won't be kind of weighed down by water. So you might be wondering, why is that beneficial? Why is it beneficial not to be weighed down by water? And to answer that question, we can actually just go ahead and do kind of a quick math problem here to kind of give some insight into that. So the math problem that I want to solve is how many grams of glycogen, including, I'm going to say this, including its water weight, because we know that all of these polar molecules attract some water, and that contributes, of course, to how much weight that they have. So how many grams of glycogen, including the water weight, will our body need, let's say, if we want to have the same number of calories? I'm going to say if we're equal to the same number of kilocalories, that the 12,000 grams of fat in the average 70 kilogram meal that we talked about above can produce. So in order to answer this question, we have the information in the table above, but we also just need to know one more thing, which is how many grams of water are associated with each gram of glycogen. And the answer to that is there's about 3 grams of water weight, so say H2O weight, associated with 1 gram of glycogen, or protein, actually, for that matter. So I'll just put that in parentheses for our reference. All right, so to answer this question, we first need to find out how many kilocalories can 12,000 grams of fat produce. And that's simply 12,000 grams of fat, right, times, from the table above, I'll remind you, we said that for every gram of fat, we can burn about 9 kilocalories of energy. And because we're just trying to get a ballpark number, I'm going to go ahead and round that up to about 10 kilocalories, okay? And that is going to be equal to 120,000 kilocalories. So that's how many kilocalories we need to be able to produce with glycogen. Now, with glycogen, we know that for every 4 kilocalories, we're able to essentially utilize, for every 4 kilocalories, we're able to get that out of 1 gram of glycogen, and that's without accounting for its water weight. But if we do account for its water weight, we know from this ratio above right here that for every gram of glycogen, there is 3 grams of water weight. I'll just put H2O here to remind us, okay? So our units are going to cancel out here, and all together, if we scroll down, we end up getting 120,000 divided by 4, ends up being 30,000 times 3, which ends up being a whopping 90,000 grams of glycogen that would weigh us down in order to produce the same number of calories as only 12,000 grams of fat. So nearly a 7 to 8 times fold difference in the amount of weight that our bodies would have to carry. And of course, this is not practical, right? Because we're talking about a 70 kilogram man, and here we have 90,000 grams of glycogen. That's equal to 90 kilograms, which is more than 100% of body weight just in glycogen alone. And so that's not practical because we haven't evolved a skeletal structure or enough muscle mass to be able to handle that extra weight. And so that's probably why fat has evolved to be the most prominent type of energy storage molecule in our body. So just to kind of summarize, we return to our list up here. We notice that not only is fat itself, just by its structure, energy rich, it's also chemically inert. It plays no functional role, unlike proteins, which are important in enzymatic function. And because fats are very hydrophobic, we're able to pack a lot in our body without carrying any extra water weight.
Wow. Wow, that's pretty cool. YouTube playback speed add-on. You can watch greater than 2 times speed. YouTube playback speed add-on. Okay. I don't know if I can. Well, I do want to understand what she's saying. If it's faster than 2x, I won't be able to hear anything.
Now, of course, there are many nutrients in food, including the imaginary cheeseburger that I've just drawn here. But in this video, I want to follow the journey of one nutrient. We're going to follow the journey of fat and how the fat contained inside of this cheeseburger is digested, transported, and stored in this imaginary man that I've drawn here. So let's say it's lunchtime, he's hungry, he takes a big bite of the cheeseburger, chews it down in his mouth, and it travels down his esophagus, which are drawn in blue here, into his stomach. It's churned a little bit more, mixed with all that acid in the stomach. And then it enters the small intestine. And the small intestine is just many, many loops repeating. And all of this surface area makes this an ideal place for all of the nutrients in our food, and of course fat is no exception to this, to be absorbed. And of course anything that cannot be absorbed will continue on in the colon, which I've drawn here in blue, and eventually you will have a trip to the bathroom. But since we are interested in how fats are digested and absorbed, we want to focus our attention here to the small intestine. So if you recall, the small intestine is lined by specialized epithelial cells, which is just a fancy way to say that there are special cells that can absorb nutrients that line the inside of this tube. And so I'm drawing kind of a representative version of one of those cells. And just to kind of orient ourselves, I will remind you that this side is the lumen of the small intestine, so the hole through which all the food is traveling. And of course, this is our cell. Now, just as for any other nutrient, the small intestine contains an array of enzymes to break down the fat molecules into smaller pieces for these cells to absorb. And as a class, these enzymes are called lipases, and some of them are secreted by the pancreas, and others are found naturally along the border of these cells. And just to kind of indicate and remind ourselves that these enzymes exist, I'm going to draw basically a Mac-Man-like character to represent these lipases that are ready to essentially break down these fat molecules into smaller pieces. But notably, these lipase enzymes must function in an aqueous environment. Remember, most of our body functions in a water-loving environment. But this, of course, poses a problem because fat molecules are very hydrophobic. So remember, when we refer to a fat molecule, we think of triacylglycerides, which I'll abbreviate as TAG here. And I won't draw the full structure, but the important things to recognize, remember, are we have a glycerol backbone, so 3 carbons here that I've shown in this kind of line diagram at the corners here, and they're attached via oxygens to 3 acyl groups, which remember are carbon double bond O, and then many, many carbon and hydrogens. And of course, some of these might have double bonds or whatnot, but the key idea here is that because we have many carbons and hydrogens, we have a very hydrophobic molecule. And for this reason, all of these molecules are not going to want to dissolve in the aqueous environment of the small intestine. In fact, they're probably going to all essentially group together and kind of want to bind with each other instead of dissolving into the aqueous environment. And so they'll kind of form these fatty droplets that won't kind of want to be broken up very much. To solve this problem, you might recall that our body secretes something called bile from the liver once food enters the small intestine. And bile is essentially a detergent for our food. So what I mean by that is if you've ever had anything greasy on your hands, you might realize that it's hard to just get that grease off by running your hand under water. But when you add soap, it's much easier to get out. And that's because soap has both a hydrophobic as well as a very hydrophilic or water-loving functional groups on its molecule. And having both of these types of functional groups allows it to essentially emulsify or essentially solubilize these fatty molecules a little bit better. So what that does is it kind of breaks up these fat molecules in kind of smaller pieces, increasing the surface area for which these lipase enzymes can act upon these fat molecules. Specifically, these lipase enzymes break down these triacylglyceride molecules by cleaving these molecules at these ester linkages by adding a water molecule across each of these bonds. And so altogether, the end result is that we form a free glycerol backbone. And now this glycerol backbone has a hydroxyl group because it's accepted a hydrogen from water, as well as now instead of acyl groups, we form what we call a carboxylic acid group because we're adding the remaining hydroxyl group from the water to these molecules. And of course, we form 3 of these. And we call these specific carboxylic acid groups, when they come from a triacylglyceride, we call these fatty acids appropriately, since of course they're coming from a fat molecule. Now at this point, these molecules are small enough to be able to diffuse into the intestinal cell. Now, although we've officially absorbed these molecules, we aren't finished, because of course we know that we need to be able to deliver these fatty acids to the tissues of our body, most notably the adipose tissue, where we can store fat for later energy use. So how do we do that? Well, the first step, once we get these molecules into the small intestine, is to turn them back to triacylglycerides, because we want to essentially pack them in a compact unit so that we can send them off to various tissues. So once we reform these ester linkages and reform these triacylglyceride molecules, we then want to package them and get them ready for transport. And the way that our cells do this is by using a carrier molecule called a lipoprotein. And essentially what this is the body essentially packages all of the triacylglycerides along with any other hydrophobic substances that the body absorbs, such as cholesterol, into the core of a protein molecule, which I'm indicating here by these purple circles. And the benefit of this is that these proteins have polar heads here. That means that they interact with the aqueous environment inside of the bloodstream, for example, but inside they're relatively hydrophobic enough to keep all of these hydrophobic molecules within them. Notably, the specific name that we call this particular lipoprotein that's produced within the intestinal cell from absorbed fats, we call this a chylomicron. So once all of these triacylglycerides are packaged into the chylomicron, the next step is for this chylomicron to leave the cell. But here's where things get a bit interesting. So let me scroll down a bit to give us some room at the bottom here. Recall that all of these cells are surrounded by capillaries, so they have a source of oxygen and nutrients, but they're also surrounded by these specialized lymphatic capillaries called lacteals. And I'll go ahead and draw one right next to it here and label that. So this right here is called a lacteal, and it's a lymphatic capillary, and this right here is just our normal, regular capillary. Now, notably, these chylomicrons are kind of big and bulky molecules, and the capillaries have very tiny fenestrations, or essentially gaps, to allow molecules to be absorbed. Now, these tiny gaps are small enough to allow capillaries to absorb proteins and carbohydrates, but they're way too small to absorb chylomicrons. And so the chylomicrons choose a different route. They are instead taken up by these lacteals, which have much larger pores relative to the capillary, and that allows these chylomicrons to travel within them. So now that we know that these chylomicrons travel through the lymphatic vessels by entering the lacteals, you might be wondering, where do they go next? And to answer this question, we just need to remind ourselves what the anatomy of the lymphatic vessels are. And the truth is, we have lymphatic vessels all over our body that drain many sites of our body. Some of them in our legs, in our thorax, some of them coming from our arms, and even some draining down from our head as well. But the key point is that most of these end up coalescing in a region that's near our neck and our shoulder. And at that point, they form ducts that empty into veins. So I'm going to just draw like a vein next to each of these 2 points here. And most of our body drains the lymph into this side on the left side, called the left thoracic duct. But some of the lymph are also drained into this right thoracic duct here as well. But the key point is that eventually anything that goes into these lymphatic vessels will drain into veins in this part of your body. And so with that brief overview of fat digestion and absorption in the small intestine, and then subsequent packaging into chylomicrons and traveling through the lymphatic vessels up to the veins in our body, we'll go ahead and take a pause and pick up and continue the journey of the chylomicron in the blood vessels in our next video.
Okay.
Recall that chylomicrons are specialized protein carrier molecules that are packaged in the small intestine with hydrophobic molecules such as fats, as well as cholesterol that we digest and absorb in our small intestine. And so shown here are representative fat molecules in yellow, surrounded by a protein shell in purple, and altogether this is called a chylomicron. Now recall that once we form these chylomicrons in the small intestine, they are packaged and then sent off from the small intestine into the lymphatic capillary, called a lacteal, next to these small intestine cells. And eventually they will drain into veins via the thoracic duct near our shoulder. And then eventually, of course, veins enter the heart, and the heart pumps it to the lungs, which reoxygenate the blood. And of course, eventually these chylomicrons will reach arteries, which drain, of course, into capillary beds, which is the site of absorption of these fats by other tissues in the body. So to demonstrate how that works, let's go ahead and clear our screen here. And we can go ahead and draw a represented, very zoomed out version of a capillary here. And so we know that capillaries are situated next to many different types of tissues. And this is how and where these tissues obtain oxygen as well as nutrients. And so we want to figure out how do the fat molecules contained within the chylomicron, shown here, how are these liberated and absorbed by the surrounding tissues? So let's say we're traveling this way down the capillary, and it turns out that there are enzymes situated in the capillary beds, which I'm going to indicate again here by using kind of a Macman-like character, because these enzymes are going to have a role of breaking some of these molecules down. And these enzymes are called lipoprotein lipase. And it's a very suggestive name. Lipoprotein refers to the fact that it's working on a lipoprotein, like the chylomicron, and the lipase refers to the fact that it serves the same function as the lipase in the small intestine. It's essentially going to take the triacylglycerides contained in the chylomicron and break them up into individual fatty acids. And the way that this occurs is that it turns out that there's a specific type of protein that's on this chylomicron, and this specific type of protein, when these lipoprotein lipases see this protein, they essentially are activated and they start breaking down these triacylglycerides into individual fatty acids and a free glycerol backbone. And so I'm not going to write the entire chemical structure, but just kind of symbols to remind ourselves of what is happening. Notably, this enzyme lipoprotein lipase is also activated by insulin, which is present in your body in response to an influx of glucose right after a meal. And because glucose and fats are often found together in foods, it seems appropriate that right after a meal, this should also stimulate lipoprotein lipase. Now, nearly all tissues can take up fats and eventually use them to extract ATP from. Notable exceptions are your brain, because fatty acids cannot cross the blood-brain barrier, as well as red blood cells, which don't contain mitochondria. And mitochondria are very important for the oxidation of fatty acids, which yields ATP. So without mitochondria, we can't extract energy from fats. But muscle, for example, can take up some of these fatty acids. And so it'll see the free fatty acids and it can take these up because they're small enough now. And perhaps the biggest user or biggest absorber of all of these fatty acids in the capillary are adipose cells. And so I'll draw a couple of representative adipose cells. And these are specialized cells for storing fat. And so they actually have very small nuclei and have just a lot of room in their cytoplasm to store fat. And so they go ahead and see that these fatty acids are floating around. And so they take up these and they eventually, to kind of compact them down for storage, turn them back into triacylglycerides and store them as kind of big fatty droplets inside of their cells. Now let's return to the chylomicron. Once it's been digested by the lipoprotein lipase, at the end of its journey through the capillary bed, we call whatever's left, we call it a chylomicron remnant, which I'll abbreviate here as CR. And of course, there might still be some triacylglycerides that weren't, broken down by lipoprotein lipase, and there's also probably some cholesterol in there that was absorbed by our diet as well. And so it still contains a lot of useful things for our body. And that is why our liver now plays a big role in reabsorbing these chylomicron remnants. So I'm just going to draw kind of a representative liver here, not really drawing anything in the right anatomical position, but just to kind of give you an overview. The liver contains specific receptors to take up these chylomicron remnants. Now you might be wondering, why the liver? Why does the liver reuptake these chylomicron remnants? Why not some other organ? And the way I like to think about the liver is that all roads of digestion lead to the liver. And so remember that while fatty acid metabolism, while the digestion of these triacylglycerides is going on in the small intestine, which I'm going to kind of just draw representatively here in this pink line here, we also have the breakdown and absorption of carbohydrates and proteins and nucleic acids as well. And these are small enough to enter the capillary bed directly. And once they enter the capillary bed, all of those capillaries eventually funnel into a vein that goes directly to the liver. And so that is a very important anatomical connection, because it means that everything that's digested and absorbed in the small intestine, except for fats, which remember are carried in chylomicrons and carried to the lymphatic vessels, pass through the liver. Now you can imagine that after a big meal, there's going to be a lot of glucose absorbed that will go through this vein into the liver. And some of this glucose will be used to make ATP, and some of it will be used to build glycogen, which remember is the main form of carbohydrate storage fuel that our body has. And it stores it directly right in this liver conveniently. But if we have a lot of extra glucose, there's actually a metabolic pathway that allows us to convert glucose into more fatty acids. Now, this is where I think the cool part really comes in. The liver has a similar functionality to the small intestine. It can package these fatty acids into triacylglyceride molecules and package them into the specialized protein carrier molecules very similar to the chylomicrons, except instead of called chylomicrons, these molecules are called VLDL, which stands for very low density lipoprotein and refers to the density of protein to hydrophobic molecules inside of it, which isn't too relevant for this discussion, but just so that you're aware of the name, this is called a VLDL particle. And so, like I said before, it contains basically a protein shell, and it packages all of those fat molecules inside of it. In addition to the fat molecules that it's packaging that have been newly made by the liver from the glucose, it also, remember, the liver was also taking up these chylomicron remnants, which also might have contained remaining fat molecules as well as cholesterol. And so both of those things can also go into the VLDL molecule. And so the liver kind of allows these two pathways to converge. Now, ultimately, just like the chylomicron left the small intestine and traveled all the way to the capillary bed so that the lipoprotein lipase could release the fatty acids, the VLDL molecule has a very similar fate. So I'm just going to put an asterisk here by this capillary bed here to remind you that the VLDL, once it reaches the capillary bed, will essentially, it can be acted upon by the lipoprotein lipase again. In addition, it also releases cholesterol to cells, but that's something that we won't cover in this video. So we've successfully followed the journey of our chylomicron remnant from our small intestine to the liver, and simultaneously how fat is transported and stored inside of our adipose cells, which I will now actually label so that we don't forget what these are. But there's one more question that we need to answer before we finish, which is what happens to the fat that's stored in these adipose cells? Now, it turns out that these adipose cells have hormone receptors on their cell surfaces, which can detect the levels of hormones that are circulating inside of the body. And this is important because remember that the major hormone that's floating around right after we've eaten a meal is insulin. But a couple hours after a meal, or even several hours after a meal, the levels of insulin begin to fall. So I'm going to write that after a meal, the levels of insulin decrease, and the levels of a different hormone called glucagon begin to increase in response to not having enough blood glucose. There are also several other hormones that are elevated during this time as well, but these are kind of the two main hormones at play. And the decrease in the level of insulin as well as the increase in the level of glucagon can both stimulate these hormone receptors. And what that does is it sends a signal inside of these cells through various modifications of different enzymes to signal these adipose cells to release all of the fatty acids from these triacylglyceride molecules into the bloodstream. To illustrate that, I'm going to go ahead and draw another kind of blown up version of a capillary bed next to all of these adipose cells to illustrate what happens after, several hours after eating a meal, in contrast to what happens immediately upon digesting a meal, which we showed on the left side here. So as I mentioned, the level of hormones signals these cells to release fatty acids. I'm just going to draw kind of representative symbols for fatty acids, and they will diffuse down their gradient because, of course, the blood now has low levels of fatty acids traveling through it into the bloodstream. And so remember, these are hydrophobic, right? And the way that the body deals with this is it allows these free fatty acid molecules to travel in and alongside these large proteins in the blood called albumin. And these are proteins that are made also by the liver. And so these fatty acids travel kind of along these proteins so that they can dissolve essentially in the aqueous environment of the bloodstream. And I just want to mention really quickly that the enzyme that catalyzes the breakdown of the triacylglycerides in the adipocytes in response to these changing hormone levels, so I'm just going to indicate that again by this Macman character here, has a special name. It's called hormone-sensitive lipase. Hormone-sensitive lipase. So again, it's a lipase because it's breaking down a triacylglyceride, and it's hormone sensitive because it responds to these changing levels of hormones. Now, returning to the fatty acids that are now traveling in the blood, most tissues in the body can now take these up. So again, we have muscle cells, my kind of bad drawing of a muscle here, and some heart cells perhaps, along with other tissues in the body. Remember, notable exceptions are the brain and the red blood cells, but most tissues can take up these fatty acids and produce a lot of ATP from them to kind of sustain them when they don't have an immediate influx of maybe fuel right after a meal. Now, finally, I want to mention that one of the biggest consumers of these free fatty acids that are floating around in the blood, thanks to hormone-sensitive lipase, is the liver. And that is because during the time of fasting after a meal, when the body needs to maintain blood glucose levels for the brain and the red blood cells, for example, that cannot use these fatty acids, the process of gluconeogenesis, or the creation of new glucose, which occurs largely in the liver, requires a lot of ATP. And the only fuel that the body has to create this ATP, the major fuel at least, are fats. And so the liver will take a lot of these fats and break them down to produce the ATP necessary to support gluconeogenesis. In addition, because we don't want to waste any of the glucose that's produced in gluconeogenesis, this process of converting glucose into fatty acids is also halted when the levels of insulin fall. And that's because this process of converting glucose to fatty acids is also stimulated by insulin. So when the insulin levels drop, this process also comes to a halt as well.
Four or 3 hours to get started on the book. Maybe. But at least that gets done. And then we'll finish it off tomorrow with the rest of Yusuf. Almost done. Yeah. Cool. Alright, I'm gonna take a 10-minute. Yeah.
So it's to be able to deliver some triacylglycerides, which I'm going to here is THC, which remember is a chemical name for a fat molecule, or free fatty acids, which I'll read here is FFA, which if you recall are the kind of monomer subunits of these fat molecules, directly into the bloodstream, where they can eventually reach capillary beds, like the one that I've drawn here. So go ahead and label this as a capillary bed. And it's important that they reach these capillary beds because it's at this point where they can diffuse to surrounding tissues such as muscle or heart tissue, for example, where they can be taken up by these tissues and oxidized to obtain cellular energy in the form of ATP. Now, I want to remind you that there are three main sources of these triacylglycerides or free fatty acids that can enter the bloodstream. And so I'm going to go ahead and scroll up here and show you kind of what I've already drawn out here and go ahead and explain it. Starting off here on the far left, I've drawn a cheeseburger, perhaps not the best drawing in the world, but just to remind us that one of our sources of fat that ultimately reaches our bloodstream is directly from our diet. So recall that our small intestine digests our food and packages the fat molecules, the triacylglycerides, into protein carrier molecules called chylomicrons, which travel through the lymphatic circulation, but eventually empty into our bloodstream where they can enter capillary beds. Now, the second way that we can get some fat into our bloodstream is directly from adipose cells. So recall that adipose cells are specialized cells inside of our body that can store large amounts of fat. And so that's kind of what I've drawn here in these circles within these cells, these large fatty droplets. And several hours after a meal, when your hormone insulin begins to drop inside of your body and other hormones such as glucagon begin to rise, they signal these adipose cells to release free fatty acids directly into the bloodstream. And recall that free fatty acids are very hydrophobic, so they kind of surf, essentially, they kind of attach themselves onto albumin molecules, which is a special type of protein that's always found inside of our bloodstream. Now, the third way that we can get some fat into our bloodstream is by synthesizing it directly inside of the liver, which I've kind of drawn an outline of here. Now, the liver cells are especially equipped with the right type and number of enzymes to be able to convert excess glucose, that is the glucose that's not being used for ATP synthesis or glycogen synthesis, into fatty acids. Then, like the small intestine, the liver essentially packages these fatty acids into triacylglyceride molecules and packages them together with cholesterol, another hydrophobic molecule, into another specialized protein carrier molecule like chylomicrons, but this one has a slightly different name. It's called very low density lipoprotein, or VLDL for short. And this, of course, is sent off to the bloodstream where it will eventually reach capillary beds and be taken up by surrounding tissues, even perhaps adipose cells, which might store it up for later use. So now that we've gone over this overview, I want to zoom in on one of these steps. I want to zoom in on this step here, going from glucose to fatty acids inside of the liver, which is commonly referred to simply as fatty acid synthesis. And to do this, I want to go ahead and kind of just zoom in on one single cell inside of the liver to visualize what's going on at the cellular level to be able to allow us to convert glucose into a fatty acid. And so I'm going to go ahead and scroll the screen here so we can have some more room. All right, so I'm going to quickly draw an outline, scroll the screen here so we can have some glucose into a fatty acid. And so I'm going to go ahead and scroll the screen here so we can have some more room. All right, so I'm going to quickly draw an outline of a representative cell, and then we'll go ahead and quickly label some important compartments that we want to talk about. So the first one is simply the cytoplasm, and there's a lot going on in the cytoplasm. But we also need to talk about what's going on in another organelle inside the cell, and that organelle is the mitochondria. And I'm going to go ahead and draw the kind of two membranes that it has here. We're not going to talk about this too much, but just because it is important to remember that this has an inner membrane and an outer membrane. Remember that the electron transport chain is, of course, located on the inner membrane. And the mitochondria is also, within it, it's the site of the Krebs cycle, which continues, notably, to break down glucose following glycolysis, which takes place inside of the cytoplasm. Now, since we ultimately want to get down to how extra glucose can be eventually converted into fatty acids, we need to actually make sure and remind ourselves how the breakdown of glucose proceeds. So as a very, very quick review, recall that glucose enters our cells from the bloodstream, and it enters the metabolic pathway called glycolysis, which takes place inside the cytoplasm. And the end product of glycolysis is pyruvate. And I'll also remind you that for every one molecule of glucose, which is a 6-carbon molecule, so 1, 2, 3, 4, 5, 6, we form 2 molecules of pyruvate, which is a 3-carbon compound. Subsequently, pyruvate is actively transported across the mitochondrial membrane by specialized carrier proteins located on the membrane. And once pyruvate reaches the inside of the mitochondria, also known as the inner mitochondrial matrix, there is an enzyme that's only found in the mitochondria called pyruvate dehydrogenase, often abbreviated as PDH, which oxidizes and removes 1 carbon from pyruvate. So remember, we had 3 carbons, and now it turns it into a 2-carbon molecule called acetyl-CoA. Now, you might recall that this 2-carbon structure is not done being broken down or oxidized. There's still some energy that we can extract from this 2-carbon molecule, and it's extracted inside of the Krebs cycle. So remember that there are many, many intermediates along the Krebs cycle, but I only want to mention a couple that will be relevant when we talk about how this breakdown of glucose converges with the synthesis of fatty acids. So remember, first off, that a 4-carbon molecule called oxaloacetoacetate, which I'm abbreviating here as OAA, combines with 1 molecule of acetyl-CoA to produce a 6-carbon molecule now called citrate. And citrate continues to be modified, oxidized, and even broken down a little bit more, and it returns to form oxaloacetate, which means that we lose 2 carbons somewhere along this cycle, which we do indeed. We lose these as 2 molecules of carbon dioxide. So there's 2 carbons of acetyl-CoA, exit as carbon dioxide, and we also form a number of reduced electron carrier molecules called NADH and FADH2, which shuttle their electrons from the oxidation process that occurs in the Krebs cycle to the electron transport chain, which is located on this inner mitochondrial membrane. How convenient, right? And then from there, we can produce ATP using oxidative phosphorylation. All right, so after that quick whirlwind tour of the breakdown of glucose, you might be wondering, where do we convert glucose into fatty acids? And it turns out that one of the intermediates of the breakdown of glucose, which is acetyl-CoA, this 2-carbon molecule located in the mitochondrial inner matrix, is a precursor for fatty acid synthesis. And we're going to go through all of the steps, but just to take a step back for a moment, the big picture way that I kind of like to think about this is that remember that fatty acids, I'm going to go ahead and draw to the side here. Remember that most of it is just a repeating carbon-hydrogen backbone, shown here in this kind of line stick model here. And so in that sense, really, we want to basically be able to link together carbon-carbon bonds. And this acetyl-CoA is just a pair of carbon-carbon bonds that we can ultimately link together. Now, it turns out that we have an interesting situation when it comes to fatty acid synthesis and linking all of these acetyl-CoA molecules together, which is that all of the enzymes necessary for fatty acid synthesis, and say enzymes for fatty acid synthesis, are located in the cytoplasm. And that's a bit problematic because remember, our acetyl-CoA molecule is in the mitochondria. Now, your first thought might be, well, pyruvate was able to shuttle across using some protein carrier molecules in these membranes into the mitochondria. Why can't acetyl-CoA do the same, going the opposite direction? Unfortunately, for some reason or the other, our body has evolved not to have any means to be be able to transport this through its mitochondrial membrane. There are no protein transporters or carrier molecules like we have for pyruvate to be able to essentially shuttle acetyl-CoA in either direction across this mitochondrial membrane. But notably, our body does have a protein shuttle across this mitochondrial membrane for the molecule citrate. And remember that citrate contains acetyl-CoA. Of course, it also contains this molecule acetyl acetate that combines with it. And so let's see what happens when this shuttles across the mitochondrial membrane. Now, once citrate reaches the cytoplasm, it turns out that there is an enzyme within the cytoplasm that is able to break citrate up back into oxaloacetate, as well as the molecule that we're interested in, which is, of course, acetyl-CoA. Now, when I first learned about this, it kind of struck me as a really roundabout way to kind of accomplish what seems like a pretty simple task, right? Which is to get acetyl-CoA into the cytoplasm where the enzymes for fatty acid synthesis can link it together to form a fatty acid. But it turns out that there might be a benefit for this citrate shuttle to make fatty acid synthesis perhaps more efficient. And so I want to briefly talk about that, but I want to erase this just to give us some more room. Now, it turns out that this 4-carbon molecule, oxaloacetate, is not going to be used for fatty acid synthesis. And so naturally, our body says, why don't we recycle it? And in fact, we do have some enzymes that can convert it back to this molecule, pyruvate. And notice that pyruvate can essentially, once it goes back to the mitochondria, it'll be turned into acetyl-CoA, and this entire cycle can continue. Now, although we're not going to go over the detailed mechanism by which oxaloacetate is converted to pyruvate, what is important, kind of a big picture idea to note here, is that we're going from a 4-carbon to 3-carbon molecule. And so we're going to lose a carbon actually as carbon dioxide during this process. And simultaneous with this step, we're actually oxidizing that particular intermediate. And so when we oxidize something, we're able to reduce something else. And it turns out that what we reduce in this case is a molecule of NAD+. And so it's reduced to NADPH. And you may recall that you've seen NADPH also as a product of the pentose phosphate pathway. And of course, we normally think about the pentose phosphate pathway being the major pathway for the production of NADPH, but this step also allows us to produce a molecule of NADPH as well. Now, one of the uses of NADPH that you might recall is that because it's associated with these electrons, it can serve as a source of reducing power to help with anabolic reactions. And remember that anabolic reactions are anything that involve building up a molecule, including fatty acid synthesis, which is exactly what we're trying to accomplish here. So to summarize and just kind of tie up everything that we've just talked about here, we've been able to get acetyl-CoA into the cytoplasm where all of the enzymes necessary for fatty acid synthesis are located. And this is important because we're going to use acetyl-CoA, multiple acetyl-CoA kind of a precursor molecules, so to say, to build up a fatty acid. And of course, because this is an anabolic reaction, we're going to need some ATP somewhere along the way. And we're also going to need some reducing power to kind of help form all of those carbon-carbon bonds. And we can get that using NADPH. Of course, NADPH can be supplied by the pentose phosphate pathway, but conveniently, perhaps, by using the citrate shuttle, we're also able to produce a molecule of NADPH from the conversion of oxaloacetate into pyruvate. In the next video, we'll pick up right here in the cytoplasm to talk about how this conversion from acetyl-CoA into a fatty acid occurs. To return to some basic principles.
Man, why are these so long?
Basic principles, and to put everything in perspective for you, I want to remind you that fatty acid synthesis is like any other anabolic reaction that occurs in our body. Remember that the common theme with anabolic reactions is that we take some type of monomer, generally, and we link these together to form a polymer. And the challenge with these types of reactions, of course, that generally these reactions have a positive delta G value, or in other words, they are not spontaneous. They require an input of energy to occur. So obviously this is a challenge, but our body resolves this challenge by coupling this reaction with a reaction that has a favorable delta G value, that is a delta G value less than 0. And in this case, our body uses the energy currency, or cell ATP. And of course, we know its reaction with water has a very favorable delta G value, very negative. And so when we break it down, we form ADP and a free phosphate group. Now, I just want to remind you here that our monomer, when we're talking about fatty acid synthesis here, is acetyl-CoA. So these two carbon subunits, we're going to link these together to form a large fatty acid chain. Now, before we get into these specific steps of fatty acid synthesis, I want to kind of take a bird's eye view first and write out what the overall chemical reaction would be for fatty acid synthesis occurring in our body. So essentially what I'm doing is I'm adding these two equations together. And just to make this super clear, I want to remind you that in order for this reaction to occur, obviously we need to have a favorable delta G value. So overall, this is going to have a negative delta G value, which tells you, of course, that the hydrolysis of ATP is much more negative than this polymerization reaction is positive. So, of course, if we add these two delta G values together, we get an overall negative delta G value. All right, so the overall chemical reaction that I want to write out for fatty acid synthesis is for the product of a 16-carbon fatty acid chain that is called palmitic acid. And palmitic acid happens to be the primary product of fatty acid synthesis in our body. So we're going to focus on this. But just so you know, our body can essentially use this kind of basic product of fatty acid synthesis to synthesize longer chain fatty acids if need be. So remember that we want to start off with our monomer, which is acetyl-CoA. I'll remind you that this is a 2-carbon molecule. So if we need to make a 16-carbon fatty acid, we would need 8 acetyl-CoA molecules. And of course, since this is an anabolic reaction, we need ATP to make an appearance as well. And it turns out 7 ATP molecules are required for this reaction. And in addition, we also actually use a molecule called NADPH. And you might recognize this from the pentose phosphate pathway, but remember that we've also produced NADPH when we've shuttled this acetyl-CoA over to the cytoplasm from the mitochondria, because remember, that was done by a molecule called citrate. And citrate broke up into acetyl-CoA as well as oxaloacetate, which the oxaloacetate produces NADPH as it was being recycled back to pyruvate. And this is really important because it's a source of electrons that will be able to reduce any oxidized groups, any groups that are oxidized on this acetyl-CoA molecule. So what I will show you in a minute is that acetyl-CoA has a carbonyl group in its molecule. And of course, we want to make just carbon-carbon bonds that are essentially attached to hydrogen only. And so in order to reduce this bond, we need a source of reducing power, and NADPH is that source of reducing power. And it turns out that we need 14 NADPH molecules. So that's all of the reactants that we require, and we form this palmitic acid. And of course, we also form the byproducts of the rest of these reactants. So we started off with 7 ATP, we end up with 7 ADP, and of course 7 free phosphate groups, and 14 NADP plus, because we've removed these electrons, and of course we're oxidizing this now, and we also end up losing water molecules as well during the course of this reaction. End up losing 6 water molecules. And I'll actually go ahead and move the 14 down here so it doesn't confuse you. And I also forgot just one more thing here. So it turns out that these coenzyme A functional groups here on this acetyl-CoA molecule will also be removed in the process of making these carbon-carbon double bonds. And so we'll also form, as a byproduct, 8 coenzyme A molecules. All right, so I'm going to go ahead and scroll down so that we can return to the cytoplasm and figure out how acetyl-CoA is polymerized into this palmitic acid. Now, because I know that it's quite easy to get kind of lost in the details, especially when talking about metabolic pathways, my goal here is not to provide kind of a detailed reaction mechanism for every step of this reaction, but rather to kind of just cover the high-yield steps, the steps that are kind of the most important, I think, for you to be able to take away. All right, so once we're in the cytoplasm and we have acetyl-CoA at our disposal, it turns out the first step of fatty acid synthesis is to charge up this molecule to a higher energy molecule called malonyl-CoA. And just in the fact that I'm saying charging up this molecule should clue you into the fact that this is thermodynamically unfavorable, and so we need something that is thermodynamically favorable to be coupled with this reaction. And indeed, that is the hydrolysis of ATP, which I'll just write shorthand here is ATP going to ADP plus a free phosphate group. Now, it might be tempting to think that we're charging up this acetyl-CoA molecule with this phosphate group here, but what we're really doing actually is we're charging it up with a carbon dioxide molecule. And the breakdown of hydrolysis of ATP is really what's fueling this reaction forward. So to give you a sense of what kind of this looks like chemically, let me go ahead and draw out the chemical structure of these two molecules. So acetyl-CoA, we begin with this kind of 2-carbon backbone structure that we call an acyl group. So carbon double bonded to oxygen and also bonded to this carbon CH3 group here is attached to a coenzyme A group via a sulfur atom here. And I'm going to leave this CoA group abbreviated like this just for simplicity. And our malonyl CoA group looks very similar. So we have a carbon double bond to oxygen. Our coenzyme A group is untouched, but we end up, plopping off a hydrogen here and essentially adding on this carbon dioxide molecule like such. And I'll go ahead and put a hydrogen here just for now, but it might be protonated or it might not be, depending on the pH of the solution, obviously. But just to kind of give you a sense here, this is what malonyl CoA looks like. Now, after this activation step, we're ready to polymerize. We're ready to put these malonyl CoA molecules together to form the 16-carbon palmitic fatty acid. So to do this, it turns out that this carbon dioxide molecule that we added to this malonyl CoA group, it plops off once again. So it might kind of seem weird to you that we're adding a carbon dioxide molecule and it plops off again. But what this is really doing, as I'll touch on in a bit, is it's making this overall reaction thermodynamically favorable, because otherwise it wouldn't be able to occur. And notably, we also happen to lose some water molecules as well in the process. And this is also where the addition of NADPH comes in, because we need the NADPH to reduce these carbon double bonds, as I mentioned earlier, so that we can create that nice string of carbon-carbon and carbon-hydrogen bonds that is what forms the bulk of our fatty acid. Now, I do want to get this stoichiometry right here and remind you that what I've written out here, this loss of one water molecule and this loss of the carbon dioxide group and the addition of these two NADPH molecules, is for each subsequent addition of these two carbon subunits. So every time we make another cycle, we're going to have another 2 NADPH and another loss of carbon dioxide and another loss of water. And that will essentially continue until we've built this 16-carbon palmitic acid chain. Before I touch more on this polymerization, let me just actually scroll down here and talk a little bit about the enzymes that are carrying out these kind of two major steps. So the first enzyme that carries out this activation step has a very nice name that's easy to remember. It's called acetyl-CoA carboxylase. So essentially, tells us exactly what it's doing. It's adding a carboxy group to this acetyl-CoA molecule. And what's important about this particular enzyme, let me scroll down a little bit more here, is that it ends up being a very highly regulated part of this entire sequence. Now, the reason this is a really good step to regulate is because it happens to be the rate-limiting step of this entire fatty acid synthesis pathway. In other words, out of all the chemical reactions that occur, this one is the slowest kinetically. And so, because the entire rate, the overall rate, how fast this fatty acid synthesis pathway can occur is limited by this one step, it makes sense that we should essentially regulate whether this step is on or off. And the way we do this is by allosteric regulation, which, if you remember, is a specific molecule binding to a non-active site portion of this enzyme to make it work better or worse, or can also regulate it with hormones as well. So I'm going to say hormonal. So I just want to mention a couple of these that are going to be relevant. So first off, we have citrate, which is an allosteric activator of this enzyme. Makes it work, makes this enzyme work better, and hence increases the flow through this pathway. Then this kind of makes sense to me because I think of the citrate shuttle that's shuttling all this acetyl-CoA to the cytoplasm. More acetyl-CoA means that we should take advantage of this acetyl-CoA and make more fatty acids, hence kind of moving this pathway forward. In terms of hormones, the hormone that activates this pathway is insulin. And this makes sense to me again because I know that right after, chowing down on that cheeseburger, we know that our levels of glucose in our blood are also going to rise. And so, if we have a lot of extra glucose around, we're going to have a lot of extra acetyl-CoA. Hence, once again, we want to move this acetyl-CoA down this pathway to produce more fatty acids. Now, in terms of inhibitors, allosterically, it turns out that fatty acids, especially, I'll say, long-chain fatty acids, can inhibit this enzyme. And the way I like to think about this is it's just kind of a form of product inhibition. If we have too many fatty acids being produced, this process is basically the cell saying, hey, acetyl-CoA carboxylase, you can slow down a little bit. We have more than we need. And in terms of hormones, the hormone that inhibits this enzyme is glucagon, as well as a couple of others, but this is one of the main ones. And the way I like to think about this is kind of twofold. So remember that glucagon levels rise several hours after a meal once we have low blood glucose levels. So if we don't have an excess of glucose, we're not going to have an excess of acetyl-CoA, and so we won't want to be forming all of these fatty acids. But I also remember that glucagon, the rising levels of glucagon also signal so remember that glucagon levels rise several hours after a meal once we have low blood glucose levels. So if we don't have an excess of glucose, we're not going to have an excess of acetyl-CoA. And so we won't want to be forming all of these fatty acids. But I also remember that glucagon, the rising levels of glucagon also signal those adipose cells to release those fatty acids into the bloodstream for all of our cells to break down to produce more ATP. So if we're in a state of kind of net breaking down of fatty acids, we don't want to be simultaneously building them back up, because ultimately we want to be able to, in this phase at least, extract the energy by breaking the fatty acids down. Now I want to mention what the enzyme is called that polymerizes these malonyl-CoA subunits together to form this palmitic acid. And the name of this enzyme is also quite easy to remember. It's called fatty acid synthase. So we're synthesizing a fatty acid. Now, as you probably saw from this arrow, there's a lot of things going on here. We're adding NADPH molecules, the carbon dioxide molecules falling off, and water is falling off too. And we're not going to go over all of the reactions that occur in this enzyme, but I think if we scroll down here a little bit, it'll be helpful to kind of just get a sense for how these carbon-carbon bonds are forming. Now, to save us some time, I've pre-drawn a couple of things that I want to go ahead and explain to you right now. So starting here on the left, what I've drawn here is the fatty acid synthase enzyme. What I want to kind of draw your attention to, obviously this isn't what the enzyme actually looks like, but a representative drawing is the fact that it has two identical subunits shown here, and they each have a thiol group or a sulfur hydrogen group located here in the active site region. So the very first step of this polymerization reaction is actually to start off with both a molecule of acetyl-CoA and malonyl-CoA. And one acetyl-CoA molecule attaches to one of those sulfur groups, and the malonyl-CoA molecule attaches to the other sulfur group, so that they're in close vicinity of one another. But I do want to point out that obviously these are no longer called acetyl-CoA and malonyl-CoA, because you can notice here that there's no coenzyme A anywhere. And indeed, when these carbons here formed a bond with the sulfur atom on these enzymes, they lost their coenzyme A group. So that's kind of where we lose our coenzyme A group right here. Now, in terms of carbon-carbon bond formation, I want us to put on our organic chemistry hats on for a moment. Now, I've already told you, up above here when we were looking at this reaction mechanism, that we lose this carbon dioxide molecule that we've added in this malonyl-CoA molecule. But how does this impact the carbon-carbon bond formation? So here I want you to notice that when this decarboxylates, when these electrons decide, hey, let me form a double bond here and let me kind of plop off of this molecule, that these electrons now make this carbon very nucleophilic. In other words, these two electrons want to bond with something. And so what they'll do is they'll see the nearest carbon that has a suitable leaving group around, and it happens to be this carbon right here because this enzyme is nicely putting us in vicinity of another carbon bond. And when it makes this carbon bond, this carbon will say, hey, I like that bond a little bit better. I'll plop off and give you my extra electrons. So what we've done essentially is we've made a carbon-carbon bond, and we've freed a space here to allow the input of another malonyl-CoA molecule. And this cycle will continue to repeat as making carbon-carbon bonds until we form the 16-carbon palmitic acid. Now, I know I haven't really covered where the NADPH comes in to reduce these carbons bonded to oxygen or where there's a loss of water, but I will actually mention that it's quite similar to the reverse of beta oxidation, which you can take a look at as well. But ultimately, after all of that is done, after we make this carbon bond, which I think is the most crucial part of this fatty acid synthesis, and we reduce those carbons bonded to oxygens, ultimately we form this 16-carbon palmitic acid, which I've drawn the structure of below. So it's a completely saturated fatty acid that we can use to make longer chain fatty acids if need be. And I want to remind you that the ultimate fate of these fatty acids, remember, they're going to be attached to a glycerol backbone to form triacylglyceride molecule, and those are going to be sent off by the liver via VLDL particles to essentially deliver these fats to the rest of our tissues.
Back annual commands. Just want to put the plate away.
I got some milk.
What I've drawn here is the chemical structure for a triethylglyceride. And recall that this chemical structure is commonly what we are referring to when we talk about the type of fat found in our food, as well as how fat is stored in our body. Now, the question I want to begin to answer in this video is how do we extract ATP, the chemical energy, from this molecule? Because you've probably heard that fats are a very rich source of energy, but how exactly do we get ATP from a structure like this? Well, the first thing I point out is that 95% of the chemical energy that we can extract from this molecule comes from these carbon-hydrogen-rich chains that we usually refer to as fatty acid chains. I'm going to put under or beside this that 95% of our chemical energy, I'll just make a symbol here, of the energy that we can extract comes from these fatty acid chains. Of course, that means the remaining 5% of chemical energy that we can extract from this molecule comes from this glycerol backbone right here. So this tiny portion of the molecule is not going to really contribute a lot. And, it essentially can enter actually glycolysis, potentially, where it can be oxidized further to produce a little bit of chemical energy. So because these fatty acid chains are contributing to the bulk of the energy that we're extracting, we're going to focus on how we extract ATP from these fatty acid chains in particular. And now to help us kind of get a bird's eye view of how we're able to extract ATP from these fatty acid chains, I've actually went ahead and drawn out a 16-carbon saturated fatty acid that our body can synthesize, which is called palmitic acid. Now, if you think back to how we extracted chemical energy or ATP from glucose, you might remember that we oxidized glucose. We essentially stripped it of its electrons and we transferred those electrons to electron carrier molecules to form reduced intermediates like NADH and FADH2, and these carried the electrons found in that glucose to the electron transport chain, where we were able to produce ATP quite efficiently. Ultimately, we just simply want to do the same thing with our fatty acid. We want to be able to oxidize it, extract all of those electrons, transfer them to those electron carrier molecules, NADH and FADH2, to be able to be used to produce ATP in the electron transport chain. And from a bird's eye view, I think the big picture takeaway is to realize that what we want to do is essentially the reverse of fatty acid synthesis. We want to be able to take this long string of carbons and hydrogens and essentially break them down into 2 carbon subunits each. And as we break them up into these 2 carbon subunits, we're also simultaneously oxidizing them to release all of this energy. And ultimately, what we're doing is we're breaking up this large fatty acid into single molecules of acetyl-CoA. And if you remember, the structure of acetyl-CoA looks something like this. So 2 carbons, 1 attached to an oxygen, and of course we have our coenzyme A group here, which I'm abbreviating like this. Now, notably, the energy extraction process doesn't stop there. Remember that acetyl-CoA is quite a versatile metabolite when it comes to metabolism. Remember that this is what can enter the Krebs cycle. So it can enter the Krebs cycle in the mitochondria, and when it enters the Krebs cycle, even more electron carrier molecules like NADH and FADH2 can be produced by further oxidizing this molecule. And so altogether, you can see that the amount of ATP that's going to be produced can be enormous because we're getting electron carrier molecules both from the direct oxidation into acetyl-CoA, as well as acetyl-CoA's own oxidation in the Krebs cycle. And to give you an idea of how much ATP we're really talking about, I've went ahead and calculated that for each run through the Krebs cycle, we can produce about a net of 10 ATP per acetyl-CoA molecule, and we're producing 1, 2, 3, 4, 5, 6, 7, 8, each of these pairs of acetyl-CoA carbons. And so altogether, we're producing about 80 ATP in the Krebs cycle alone, and then add that to the amount of ATP that's produced in this direct oxidation into acetyl-CoA, and that happens to be about 27 ATP. And I've calculated these numbers based on counting up how many NADH and FADH2 molecules are produced at each step, and then multiplying that by a conversion factor. And the commonly accepted conversion factor is that they're about 2.5 ATP per molecule of NADH produced, and 1.5 ATP per FADH2. But the big point that I really want to drive home here is that in the end, ultimately, we're producing 80 plus 27, which is 107 ATP molecules in total just from the oxidation of one 16-carbon fatty acid. Now compare that to the amount of ATP that we produce from one molecule of glucose. One molecule of glucose gives us about 30 to 32 molecules of ATP. So that's per one molecule of glucose. So you can see here how much more ATP that we can extract from this fatty acid.
Yeah.
What I've drawn out here are three compartments found inside of a cell. And what I want to go ahead and do now is kind of label what I've drawn out here, because these three compartments are going to be very important when discussing the three kind of major phases involved in oxidizing and extracting ATP from fatty acids inside of the cell. So starting up here, this compartment I'm going to label as the cell's cytoplasm. And these two lines down here are both representing the two membranes around the mitochondria. So if you remember, the mitochondria has two membranes. It has the outer mitochondrial membrane facing the cytoplasm, and then below it, it has the inner mitochondrial membrane, and I'm abbreviating these as OMM and IMM. And so, of course, the space between these is the intermembrane space. And if you remember, this is where the proton gradient was built up for the electron transport chain. And then finally, down here, this is going to be the inside of the mitochondria, which is something that we also commonly refer to as the mitochondrial matrix. Now, you might recall that fatty acid synthesis takes place inside of the cytoplasm, where the enzymes required for that process are located. Now, on the other hand, it turns out that the enzymes required for the oxidation of fatty acids to obtain all of that ATP from that molecule are located inside the mitochondria. But remember that cells take up fatty acids into their cytoplasm from the bloodstream. So we need some way to be able to transport these fatty acids from the cytoplasm into the mitochondrial matrix. And this might look familiar to you. It might kind of ring a bell from fatty acid synthesis, because remember, in fatty acid synthesis, we were also having to transport something across the cytoplasm. Of course, we were going the opposite direction in fatty acid synthesis, where we wanted to transport acetyl-CoA into the cytoplasm. But I actually want to come back at the end of the video to touch on kind of the seemingly roundabout way to have to transport things across this mitochondrial membrane, because it turns out it has a very interesting implication for how our body is able to regulate fatty acid synthesis and oxidation. And so I'll come back again and touch on that. But just going back to this transport process, it turns out that in order to be able to transport these fatty acids across this mitochondrial membrane, there's a specific pathway that requires us to actually activate, so to say, a fatty acid molecule with another molecule in order for this transport machinery to work. And so altogether, there are kind of three major phases involved in being able to ultimately extract ATP from a fatty acid. So let's go ahead and cover these three phases in turn. All right, so starting off with the activation step, let me go ahead and draw out the chemical structure of a fatty acid, starting off with its carboxylate head group. And of course, it also has a long chain of carbons and hydrogens that comprise its fatty acid tail, which we usually refer to it as. But instead of drawing this tail out every single time or drawing or writing out all the carbons and hydrogens, I'm just going to abbreviate that using the letter R to keep things in our diagram simple. Now, as I mentioned before, the goal here is to activate this molecule, quote unquote, with another molecule so that we're able to transport it into the mitochondria. So how does the body do that? Well, the body does that by reacting with fatty acid with one of the most versatile metabolites in our body, which is coenzyme A. Because the structure of this molecule is quite large, we end up just abbreviating it as CoA, and usually most textbooks will highlight this one functional group, this sulfur hydrogen group, this thiol group, because it's involved in forming a bond with this carbon here. So let's kind of see what that looks like, what the product of this reaction is. So we end up preserving this acyl group, and the acyl group just refers to this kind of part of the molecule here. We end up forming a bond with the coenzyme A through the sulfur atom as such. And the name of this molecule is acyl-CoA. And I remember when I was first learning about this, I confused this often with acetyl-CoA, which remember just has a methyl group instead of this long fatty acid chain. So just keep those two things in mind, making sure not to confuse this acyl-CoA with acetyl-CoA. Now, as a quick disclaimer, I want to apologize in advance for any stoichiometry calculations that don't seem to be right, perhaps like this oxygen atom here. Where is this oxygen atom going? And that's due in part because I'm abbreviating some things in these molecules, and so the oxygen atoms might be hidden somewhere, but mostly it's due to the fact that I'm not going over the entire mechanistic pathway by which this reaction occurs. It ends up there are a couple steps involved in this reaction. And so I encourage you, if there's something that doesn't make sense in terms of stoichiometry to just do a quick Google search and the entire mechanism will be illuminated. But I'm just trying to give you the big picture and keep our diagram a little bit neater here by just kind of giving you the big picture here. And so going back to this particular reaction, it turns out that like any reaction where we have to activate something with a higher energy functional group, perhaps so to say, we need some input of chemical energy. And indeed, this reaction is coupled to the hydrolysis of ATP. And in fact, ATP, we normally think about it as ATP going to ADP in a free phosphate group, but in this case, we actually go all the way to a monophosphate group and produce what we call a pyrophosphate group, which is just two phosphate groups stuck together. And what really makes this reaction thermodynamically favorable, just kind of as a fun fact here, is that it turns out that when this reacts with the water, it splits up into two individual phosphate groups. And this hydrolysis reaction right here ends up having a very negative delta G value. And so that's kind of what drives this overall reaction forward. So just to summarize here, we've successfully transformed our fatty acid into an acyl-CoA group, which is what we refer to as an activated fatty acid for the transport process that we'll talk about next. But before we do that, I do want to mention that the enzyme that catalyzes the reaction is actually located on this outer mitochondrial membrane here, and it's called acyl synthetase. So I kind of just think that we're synthesizing essentially another acyl group, an acyl-CoA, acyl synthetase. That's kind of how I try and remember it by. All right, so what happens to our acyl-CoA molecule next? Well, it turns out that there is another molecule inside of a cell by the name of carnitine, and this usually has a kind of bulky chemical structure to draw out. So I won't draw it completely, but what I will draw out is the fact that it has a hydroxyl group here. I'm drawing this hydroxyl group as oxygen bound to hydrogen because if I put on my organic chemistry hat for a moment, I remind myself that this oxygen can serve as a nucleophile and form a bond with the carbon on this acyl-CoA molecule like such. And the coenzyme A group that we added can serve as a leaving group, and the sulfur essentially can take back its electrons. And so ultimately, what we've done is we've, to this carnitine molecule, which I'll abbreviate here as just C now for simplicity, we've attached via this oxygen right here our fatty acid. And so we have a structure that looks something like this. Now, just as a quick aside, one way that I kind of remember the name of this molecule and its function in fatty acid oxidation is that I kind of think about it as being carnivorous, carnitine being carnivorous for the fatty acid. So it kind of essentially takes a bite into this acyl-CoA glycogen right here, which I'll abbreviate done. It takes back its electron-CoA molecule like such. And the coacyl synthetase, that's kind of how I try and remember it by. All right, so what happens to our acyl-CoA molecule next? Well, it turns out that there is another molecule inside of a cell by the name of carnitine, and this usually has a kind of bulky chemical structure to draw out. So I won't draw it completely, but what I will draw out is the fact that it has a hydroxyl group here. And I'm drawing this hydroxyl group as oxygen bound to hydrogen because if I put on my organic chemistry hat for a moment, I remind myself that this oxygen can serve as a nucleophile and form a bond with the carbon on this acyl-CoA molecule like such. And the coenzyme A group that we added can serve as a leaving group, and the sulfur essentially can take back its electrons. And so ultimately, what we've done is we've, to this carnitine molecule, which I'll abbreviate here as just C now for simplicity, we've attached via this oxygen right here our fatty acid. And so we have a structure that looks something like this. Now, just as a quick aside, one way that I kind of remember attached via this oxygen right here our fatty acid. Quick aside, one way we have a structure that looks in fatty acid oxidation is that I kind of think about it as being carnivorous, carnitine being carnivorous group, and is able to attach it to itself like this. So just a quick aside in case that helps you remember anything. But going back to this particular reaction, this reaction also has an enzyme that carries this out, and it's called carnitine acyl transferase. And it's located on, again, the outer mitochondrial membrane. And so I'll go ahead and write that out. So carnitine acyl transferase. So pretty logical name, right? It's transferring the acyl group onto the carnitine molecule. And it's actually denoted as carnitine acyl transferase 1 because we will need another one of these enzymes on the inside of the mitochondrial matrix in just a bit. So keep that at the back of your mind.
Tell me to read the book English.
Now, it turns out that relative to the inner mitochondrial membrane, the outer mitochondrial membrane is a bit more permeable to many molecules. Now, it turns out that relative to the inner mitochondrial membrane, the outer
Okay.
Now, it turns out that relative to the inner mitochondrial membrane, the outer mitochondrial membrane is a bit more permeable to many molecules.
Am I losing an inner or something? Oh, okay, I'm back.
Now, it turns out that relative to the inner mitochondrial membrane, the outer mitochondrial membrane is a bit more permeable to many molecules. And specifically, it contains specialized proteins called porins that have essentially created little tunnels or pores for molecules to kind of diffuse through. So I'm kind of just drawing a representative pores to remind us of that fact. And it turns out that this carnitine attached to this fatty acid, which is actually commonly referred to now as an acyl carnitine molecule, can readily diffuse from the cytoplasm into the intermembrane space here via one of these pores. And it's actually a bit unclear to scientists whether it's the carnitine and the fatty acid, the acyl-CoA, that both diffuse into the intermembrane space and then get kind of attached via this carnitine-acyl transferase, or whether it's more like what we've drawn up here, and that everything occurs in the cytoplasm and this entire acyl carnitine molecule diffuses into the intermembrane space. So I just wanted to mention that, but in any case, the ultimate endpoint is the same. We get this acyl carnitine into this intermembrane space via these pores in the outer membrane. Now, at this point, you might be wondering to yourself, why did we go through all the trouble to create this acyl carnitine molecule if these porins are permeable to a lot of things, including even fatty acids? So why can't the fatty acids just go through these porins and get into the mitochondrial matrix? Well, first, I want to remind you that, remember, we're only halfway home. We have to still traverse this inner mitochondrial membrane in order to get to the mitochondria. And it turns out that this inner mitochondrial membrane does not have these nice pores in its membrane, so it's far less permeable to molecules in this outer mitochondrial membrane. But what it does have is it has this protein transporter within its membrane that's going to help us out a little bit. And this protein transporter is called acyl carnitine. So already it's giving you a clue that it's going to recognize that molecule, acyl carnitine, that we worked so hard to create. And it's called acyl carnitine translocase. So just like its name suggests, it's going to essentially translocate this acyl carnitine from this intermembrane space into the mitochondrial matrix. And to show that, actually, I'm going to go ahead and take a quick shortcut here and actually go ahead and copy and paste this molecule here from the cytoplasm to the mitochondrial matrix right here. And I'll go ahead and extend our path right here from the intermembrane space, facilitated by this protein transporter, into the mitochondrial matrix. Now, of course, we're not finished here because we don't want to oxidize this entire molecule. We just want to oxidize this fatty acid chain to obtain some ATP. So we need some way to remove this fatty acid from this carnitine molecule. So remember how I said that we also have a carnitine acyl transferase on our inner mitochondrial membrane? Well, that's where this enzyme comes in. So I'm going to go ahead and abbreviate that now as CAT for carnitine acyl transferase. I'm going to put the Roman numeral 2 to indicate that it's a different enzyme located on the inner mitochondrial matrix. And what this enzyme does is it catalyzes essentially the reverse reaction of what happened inside of the cytoplasm. So what I mean by this is that it takes a coenzyme A molecule from the mitochondrial matrix, and in a reverse reaction, now our sulfur atom here is going to serve as our nucleophile essentially, and essentially displace this bond that was formed earlier, so that we end up forming a carnitine molecule back to its original form with a hydroxyl group. And we also end up forming back again simply our acyl-CoA group. So remember, that looked exactly like it does above, with our acyl group attached to an acetyl-CoA molecule via the sulfur atom. And in fact, just to make that crystal clear, I'll go ahead and actually write out the full name below it. So recall that this is the acyl-CoA molecule. Now, before moving on to the oxidation step, I want to point out one more thing, which is that it turns out that this acyl carnitine translocase protein transporter is actually quite efficient. because for every acyl carnitine molecule that it pumps into the mitochondrial matrix, it actually exchanges it for one molecule of carnitine. So this carnitine molecule that's produced by this carnitine acyltransferase 2 enzyme can actually return via this protein transporter into the intermembrane space. So I'll go ahead and continue its journey in the intermembrane space, and then out through the outer mitochondrial membrane and back into the cytoplasm, where it can be recycled to help transport another fatty acid into the mitochondrial matrix. That's kind of a pretty cool mechanism, I think. All right, so now that we've finally gotten our fatty acid into the mitochondrial matrix in the form of this molecule acyl-CoA, what happens to this molecule next? Well, using the enzymes found in the mitochondrial matrix, this molecule undergoes actually a cycle of repeating steps. And each cycle consists of 4 main steps. And at the end of each of these 4 steps, we end up producing one molecule of acetyl-CoA. And I'll remind you the structure of this molecule is a 2-carbon structure, an acyl group attached to a coenzyme A like such. And each time we also produce an acyl-CoA chain that is now 2 carbons shorter than it was before. Of course, that's because it's losing those 2 carbons and producing this acetyl-CoA molecule. I'm not going to go in depth into these 4 steps here or the enzymes used to complete these steps. But what I do want to point out big picture are the major inputs and outputs of these 4 steps to get to this molecule of acetyl-CoA and a fatty acid chain that's 2 carbons shorter. So remember that the production of acetyl-CoA involves oxidation. So oxidation, we're losing electrons, which means we need the help of electron carrier molecules to accept those electrons so they can shuttle them to the electron transport chain to produce some useful energy in the form of ATP. So remember, our 2 electron carrier molecules are FAD and NAD. And of course, these are in their oxidized form. So when they accept electrons, they become reduced into FADH and NADH. And of course, it also produces a proton as well. And the kind of convenient point that I want to point out to you is that it turns out, remember, that the electron transport chain is not far away. The electron transport chain is located on the inner mitochondrial membrane, right? So that's quite a quick journey. And so these are readily reoxidized to their oxidized form. Once they donate to the electron transport chain. And so the cycle can continue quite conveniently in this mitochondrial matrix. Now, as a couple of minor points, remember that we need some source of oxygen to be forming all of these carbons bonded to oxygen in each of these subsequent acetyl-CoA molecules that we produce. So there's actually an insertion of water somewhere along these 4 steps, as well as an insertion of an additional coenzyme A molecule for each subsequent acetyl-CoA group that's formed. Now, before we wrap things up and briefly talk about the regulation of this oxidation process, I just want to mention something that I didn't have quite the chance to talk to you in depth to you about, but something you might hear when referring to this process, which is the statement of beta oxidation. And it sounds super intimidating, but it's actually quite simple. And so I just want to take a minute to just kind of explain this to you briefly, like what the big idea behind this is. So this beta oxidation simply refers to the position of the carbon in this fatty acid chain that's being oxidized. So remember, the carbon that's being oxidized is a C double bond O bond right here in this acetyl-CoA molecule. And so to explain this to you, I'm going to go ahead and actually, I know it's been abbreviating this fatty acid chain using the letter R, but I just want to draw in a couple of these carbon-carbon bonds here. So I'll draw in a couple of CH2 groups attached to the carboxylate group here. So it turns out that the way that chemists label these carbons is based on their position to this carboxylate group here. So the carbon closest to this carboxylate group gets the designation of being the alpha carbon, and the next furthest one away is called the beta carbon. And of course, if you're further away, you also get other names as well. But just because we're talking about beta oxidation, I'll stop there. And so you can see that it's this carbon, it's beta carbon. So in other words, every other carbon relative to this carboxyl carbon here that is going to be turned into the C double bond O to form this acetyl-CoA molecule. So that in a way is probably pretty intuitive, but this beta oxidation is just kind of a fancy way to refer to that. All right, so we're hitting the home stretch here, and the only other thing that I want to mention is how this oxidation process is regulated. And it turns out that among all these reactions, the rate-limiting reaction is catalyzed by this enzyme carnitine acyl transferase. I'll actually go ahead and write that up. This is the rate-limiting step, and remember that means that kinetically it's the slowest among all of these reactions. And also remember that that's important to know when we're thinking about regulation, because since the rate-limiting step determines the overall rate of this entire reaction, it's a good step to regulate, essentially to turn on or off. And it turns out that in order to figure out how this is regulated, we need to remind ourselves a little bit about fatty acid synthesis. I'm going to remind you of a molecule called malonyl-CoA. And if you remember, this molecule was kind of a charged up version of acetyl-CoA that was used to synthesize fatty acids inside of the cytoplasm. And it turns out that this molecule is actually an allosteric inhibitor of this enzyme. So I'm going to kind of just squeeze this arrow through to indicate with a red line and a minus sign here that this inhibits this enzyme carnitine acyl transferase 1. So how can we think about this? Well, the way I kind of reason it in my head is if we have a lot of malonyl-CoA lying around in our cell, it means that we're making a lot of fatty acids. If we're making fatty acids, if we're synthesizing them, then we don't want to be breaking them down. And so we can essentially inhibit the rate-limiting step of this oxidation process to make sure that we have a net production of fatty acids. Now, another cool thing that I think is worth noting about this type of regulation is that by using a substrate in fatty acid synthesis to regulate an enzyme in fatty acid oxidation, the body has essentially made these two processes mutually exclusive. So when one is on, so when there's a lot of malonyl-CoA, the other, being oxidation, will have to be kind of essentially turned off, right? And so that's exactly what the body wants. It doesn't want to be in a gray zone. It either wants to be essentially producing a net amount of fatty acids for storage, or it wants to be breaking them down to extract all of that ATP from. And then the last point that I would make is that even though it kind of seems like a pain to traverse through all of these compartments to get to where we need to go, having these compartments makes this kind of regulation also pretty cool, because by kind of regulating this oxidation at this step right here, which is the key step for transporting the fatty acids from the cytoplasm to the mitochondria, we can essentially keep these reactions separate from one another, literally. So we're actually keeping them in separate compartments, which is a great way to regulate whether something is on or off.
Yeah.
In this video, I want to explore the question of how does our body adapt to periods of prolonged starvation. So in order to answer this question, I actually think it's helpful to remind ourselves first of a golden rule of homeostasis inside of our body. So in order to survive, remember that our body must be able to maintain proper blood glucose levels. I'm going to go ahead and write, we must be able to maintain glucose levels in our blood. And this is important even in periods of prolonged starvation because it turns out that we need to maintain glucose levels above a certain concentration in order to survive, even if that concentration is lower than normal. This, of course, brings up the question, well, how does our body maintain blood glucose levels? So let's go ahead and answer this question by starting off small. Let's say we have a mini case of starvation, let's say 3 or 4 hours after a meal. Your blood glucose levels begin to drop. And so what does your body do to resolve that? Well, at this point, it has a quick and easy solution. It turns to its glycogen stores in the liver. Remember that our body stores up these strings of glucose inside of our body so that we can easily pump it back into the blood when we're not eating. But unfortunately, humans only have enough glycogen stores to last us about a day. So after a day of starvation, our body is pretty much reliant exclusively on the metabolic pathways involved in gluconeogenesis, which, if you remember, is the pathway by which we produce new or neoglucose. And we produce this glucose from non-carbohydrate precursor molecules. So let's think about what else we have in our body. Remember that our other two major storage fuels are fats, and we usually think about fatty acids containing most of that kind of energy. And we also have amino acids. So I want to talk about both of these fuels in turn and how and if they can contribute to gluconeogenesis. Now, even if you forget most of what I say in this video, try and remember just this one next key point, which is that the breakdown products of fatty acids cannot be converted for the most part into glucose, so they can't contribute to gluconeogenesis. On the other hand, the breakdown products of the catabolism of amino acids can. So to talk about why this is the case, I want to remind you of the Krebs cycle and gluconeogenesis and how these two cycles are connected. Remember that the major breakdown product of the oxidation of fatty acids is a molecule called acetyl-CoA. And this is a very special molecule because it represents the entry point into the Krebs cycle. Remember that it combines with a 4-carbon molecule called oxaloacetate, which we usually abbreviate here as OAA, and it is turned into this molecule called citrate. And of course, there are many, many other conversions that I'm not going to write, but the gist of the Krebs cycle is that we end up cycling back to oxaloacetate, which means that we lose those 2 carbons that we added in the form of acetyl-CoA into carbon dioxide. All right, so fairly simple enough, right? We end up producing these acetyl-CoA molecules from the oxidation of fatty acids, which cycle through this Krebs cycle, allowing us to produce all of those electron carrier molecules like NADH and FADH2, which end up contributing to the production of ATP in the electron transport chain. But how does this all connect up with and implicated with this gluconeogenesis process? So let me remind you that generally, remember, we think about gluconeogenesis as essentially being the reverse of glycolysis minus a couple of steps that we have to change around a little bit because some steps in glycolysis are irreversible.
Yeah.
But largely, we can think about it as the reverse of glycolysis. And remember that the end product of glycolysis was this molecule called pyruvate, right? And so somehow we can funnel things into pyruvate. And if we end up getting this molecule pyruvate through the process of gluconeogenesis, we can actually convert it to a molecule called oxaloacetate, which should ring a bell because remember it's also in this Krebs cycle here. And then we can also then eventually turn it back into phosphoenolpyruvate, another molecule also found in glycolysis. And then we can essentially run this entire sequence of glycolysis pretty much backward with changing around a couple of enzymes, of course. And eventually we get back to producing glucose. All right, so now here's the reason why amino acids can contribute to this pathway of gluconeogenesis, but fatty acids can't. It turns out that the catabolism, the breakdown of amino acids, allows these breakdown products to be turned into some of the intermediates along the Krebs cycle. So I'm just kind of drawing arrows loosely because I know I haven't written out the specific names of the molecules, but suffice to say the big picture is that we're able to get these amino acids converted to these intermediates. And what's really cool is that we know that this molecule oxaloacetate is eventually what these intermediates are going to become. And we also know that oxaloacetate is a compound in gluconeogenesis. And in fact, we can even shuttle this more directly to be converted right into phosphoenolpyruvate right here. And so that's how these amino acids eventually get into this gluconeogenesis pathway. Likewise, amino acids can also, when they're broken down, also be converted into pyruvate as well, which from this diagram I've shown you also allows them to contribute to this production of glucose. Now, even though all the ATP that's produced by this acetyl-CoA molecule from the oxidation of fatty acids might be fueling this anabolic reaction of gluconeogenesis, because remember, anabolic reactions require ATP, this acetyl-CoA molecule in itself has no place to enter this gluconeogenesis pathway. Remember that pyruvate can be converted to acetyl-CoA using the enzyme pyruvate dehydrogenase, But the thermodynamics of this chemical reaction, the delta G value for this reaction is very negative. And so this forward reaction is irreversible. In other words, just to make this super clear, we cannot go the opposite direction. We cannot go from acetyl-CoA to pyruvate. And so acetyl-CoA cannot enter the gluconeogenesis pathway. Now, I remember when I was first learning about this, that part made sense. But what didn't make sense to me was how acetyl-CoA, it looked like acetyl-CoA could eventually be converted to oxaloacetate. And then from here, it seemed to me that we could enter the gluconeogenesis pathway right here. But I want to remind you that, remember, the two carbons that were in the acetyl-CoA molecule here, we end up losing those two carbons in these molecules of carbon dioxide. So even though our diagram looks a bit misleading in that we can kind of follow acetyl-CoA's journey to oxaloacetate, you just have to remember that you know that these two carbons, we end up not having a net production of carbons anywhere along the cycle because these two carbons end up exiting as carbon dioxide molecules. Now, a couple minor points that I do want to mention are that it turns out that there are some fatty acids, specifically odd chain fatty acids, that can contribute in some way to the Krebs cycle, like these amino acids. So some portion of these odd chain fatty acids can be converted to intermediates in the Krebs cycle and be used for gluconeogenesis. But on the whole, we generally consider fatty acids not to contribute to gluconeogenesis because we think about acetyl-CoA being the primary product of fatty acid synthesis. And the next point that I actually want to make as well is it turns out that there are some amino acids that can also be converted directly into acetyl-CoA. And so I'd ask you the question, would these amino acids be able to contribute to gluconeogenesis? So think about that for a moment. Now, of course, the answer to that question is, as long as those amino acids don't have any way to contribute to any of these intermediates in the Krebs cycle, it's just like the situation for fatty acids. Because we're ultimately turning into acetyl-CoA, there's no way for acetyl-CoA to contribute to this pathway of gluconeogenesis. So these amino acids, if they're exclusively being turned into acetyl-CoA, cannot contribute to gluconeogenesis. All right, so this is all fine and good. We have a solution, right? We're able to use amino acids to eventually produce glucose through gluconeogenesis. But what might be a potential pitfall of using amino acids? Because remember that proteins are vital molecules inside of our cell. Many of these proteins have functions inside of our cells. Many of these are enzymes that carry out important processes, and proteins often comprise the bulk of our muscle. So you can imagine that using all of these amino acids and breaking them down would be bad for our survival as a species because, of course, we need our muscle mass to be able to move around, and we need our enzymes to be performing their activities inside of our cell. So that's why in states of prolonged starvation, our body comes up with another golden rule, which is to save our protein. We don't want to waste our protein in the production of glucose. And luckily for us, many of the tissues in our body that rely exclusively on glucose for the production of energy, such as the brain, because remember, fatty acids can't cross the blood-brain barrier, many of these tissues tend to be a little bit more flexible for us in times of starvation, and they start using a different fuel that our body switches to making several days after starvation called ketones. And for the brain in particular, these ketones are effective, unlike fatty acids, because these ketones are water-soluble enough to cross the blood-brain barrier and allow us to produce ATP in times of starvation. All right, so what's the big picture idea behind ketones, and how does ketone synthesis save our proteins? So to describe this, I want to go ahead and make some more room here to draw out a flow chart to remind you what happens when our body is subjected to starvation. Remember that our hormones are the orchestrators of all metabolic pathways, and so let's remind ourselves what happens to our hormones in times of fasting. Remember, glucagon levels begin to rise because they're sensing we have low blood glucose in our blood, and insulin levels, which only respond, of course, to high blood glucose levels, begin to drop. And this stimulates our body to start breaking down fatty acids in fatty acid oxidation, which occurs primarily in the liver. And as we talked about above, this ultimately leads to having a lot of acetyl-CoA molecules. And in the beginning, about maybe 3 to 4 hours after our last meal and up to a day, our body is working on building up these acetyl-CoA stores so that they can enter the Krebs cycle to eventually produce the electron carrier molecules that will allow us to produce ATP in the electron transport chain. So that's kind of our main goal in these first kind of 1 or 2 days of fasting. However, about 2 to 3 days after fasting, the levels of acetyl-CoA inside of your body begin to rise above the amount that you need to maintain ATP levels. And the reason why this is able to happen is for several reasons. One thing to recognize is that most metabolic pathways we talked about have some form of regulation, some form of product inhibition, but it turns out that there is no type of product inhibition when it comes to fatty acid oxidation. So normally we might think that rising levels of acetyl-CoA would signal somehow the body to decrease fatty acid oxidation, but this does not occur. There is no type of product inhibition. So acetyl-CoA levels can continue to rise essentially. In addition, remember that one of the biggest regulators of metabolic pathways in our body is ATP. And if we have enough fatty acid oxidation to produce enough ATP in our cells, and ATP levels are plenty, it'll essentially feed back onto the electron transfer chain to say, hey, slow down. And if the flux through the electron transport chain is slower, then that will lead to an increase in the amount of our reduced electron carrier molecules, like NADH, for example, because these won't be donating electrons into the electron transfer chain as fast. And remember that NADH is one of the biggest regulators of the Krebs cycle. And so if we have a lot of NADH, it will slow flow through the Krebs cycle. And so the acetyl-CoA molecule will no longer want to enter the Krebs cycle as much. And that is what allows our body to shunt this extra acetyl-CoA that develops after about 2 or 3 days of starvation into the production of ketones. And this occurs mainly inside of the liver. Now, of course, just to bring the story full circle, the big idea here is that these ketones can then leave the liver, go into the bloodstream, and other tissues like the brain can take out these ketones and convert them back to acetyl-CoA. And of course, we know then that acetyl-CoA can enter the Krebs cycle like this and contribute to the production of ATP. Moreover, as the body shifts several days after starvation to this new fuel of using ketones, it puts less pressure on the body to undergo gluconeogenesis. And if there's less pressure to undergo gluconeogenesis, there will be less pressure for our body to break down protein. And so actually it turns out that the majority of protein breakdown occurs in the first 2 to 3 days of starvation. But after that, protein degradation, the rate of protein degradation will decrease because our body is shifting to use more of these ketones as an energy fuel.
Oh. Oh my goodness. Wow. Wow. We got it from the book. Okay, nice. Very cool.
In this video, I'll provide you with a crash course overview of amino acid metabolism. And specifically, I want to focus on the catabolism of amino acids and how that catabolism allows us to produce ATP inside of our cells. Now, compared to carbohydrate catabolism and fatty acid catabolism, recall the pathways of glycolysis. In this video, I want to provide you with a crash course overview of amino acid metabolism. And specifically, I want to focus on the catabolism of amino acids and how that catabolism allows us to produce ATP inside of our cells. Now, compared to carbohydrate catabolism and fatty acid catabolism, recall the pathways of glycolysis and fatty acid oxidation. Compared to those pathways, amino acid metabolism only accounts for about 10 to 15% of our cells' total energy production. So that's why I think that amino acid metabolism doesn't usually get its fair share of airtime compared to processes like glycolysis and fatty acid oxidation. And to do that, let's go ahead and follow what happens to amino acids in the fed as well as the fasted states of our body. Now, fed refers to our body's state right after, immediately after eating a meal. And remember that in terms of hormones, the hormone that's going to be elevated is going to be insulin, which is elevated in response to higher blood glucose levels immediately following a meal, and levels of the hormone glucagon are going to be decreased. Now, of course, it's going to be opposite several hours after a meal, which we call the fasted state, in which the levels of insulin will be decreased. And of course, in response to low blood glucose levels, the levels of glucagon in our body will start to rise, along with a couple of other hormones as well. But these are the two at least big hormones that regulate the bulk of metabolism in our body. Now, starting with the fed state, let's start at the beginning of the story. Recall that we ingest proteins from our food, and those proteins are broken down into amino acids inside of our small intestine. And just as a side note, you might hear the terms essential and non-essential amino acids used, especially in medical literature. And what this simply refers to is that essential amino acids are those amino acids of the 20 that we know of that our body cannot synthesize. And so we must somehow get these in our diet. Whereas non-essential amino acids can be actually synthesized in our body and we don't need them as part of our diet. But getting back to these amino acids, once they're broken down in the small intestine, they travel via the bloodstream directly to the liver, just like glucose. Now, once the amino acids have made it to the liver, several things can happen. The liver can use these amino acids directly for protein synthesis, but it can also use any excess amino acids and convert these into glucose and or fatty acids. And of course, recall that the ultimate storage forms of these two molecules are going to be glycogen in the case of glucose, which is stored in the liver mainly. And for fatty acids, we store these as triacylglycerides in our adipose tissue. So how does this conversion from amino acids to glucose and fatty acids happen, you might ask? Well, remember that the precursor for glucose, or I should say precursors, can be pyruvate as well as oxaloacetate. And for fatty acids, the main precursor for fatty acid synthesis is the molecule acetyl-CoA. And as a relevant side note, I want to point out that acetyl-CoA happens to be in equilibrium with another molecule in the cell called acetyl-CoA. And oxaloacetate, if you remember, is in equilibrium with a lot of the intermediates of the Krebs cycle. So I'm going to abbreviate here as intermediates of Krebs cycle. And there are numerous molecules with numerous names that I won't mention here, but just so that you get the big picture. Now, the key point here is that amino acids, specifically the carbon backbone of these amino acid molecules, can be interconverted and metabolized directly into the molecules and the precursor molecules that I've listed here for fatty acids and glucose. So they can be converted directly into pyruvate, into oxaloacetate, as well as intermediates of the Krebs cycle, acetyl-CoA, as well as acetyl-CoA. Now, another classification that you might hear with regard to amino acids is whether an amino acid is so-called a ketogenic amino acid or whether it is a glucogenic amino acid. And that simply refers to whether the carbon backbone of these amino acid molecules feeds into the precursor molecules for glucose synthesis or whether it feeds into the precursor molecules for fatty acid synthesis. So in this case, ketogenic amino acids are converted to acetyl-CoA or acetoacetyl-CoA and ultimately fatty acids, whereas glucogenic amino acids feed into pyruvate, oxaloacetate, or intermediates of the Krebs cycle. Now, just as a fun fact, it turns out that there are two amino acids that are exclusively ketogenic, and those are lysine and leucine. So anytime you ingest lysine or leucine, you will definitely be making fatty acids from those amino acids if they're ingested in excess. Of course, other amino acids can actually contribute to glucogenic pathways, and some might even contribute to both, but that's just kind of a fun fact. Now, going back to the journey of our amino acids here, remember that it enters the liver, and the liver can either use it for protein synthesis or convert it into other energy storage forms, but it can also send it off, and it can send it off to other tissues such as the muscle, for example, where the muscle can use it for its own protein synthesis. So other cells will also receive amino acids that are digested that they can use for protein synthesis as well. Now, moving on to the fastest state, I'm also going to put the liver here at kind of the center of our diagram, because remember, the liver is quite a centerpiece when it comes to metabolism. A lot of things are going on in the liver, and specifically in the fasted state, you might recall that fatty acids are being released from adipose tissue and being sent to the liver, where they're being oxidized, and all of that ATP is fueling the synthesis of glucose. And if the person is in a very severe state of starvation, let's say they haven't had a meal for 2 or 3 days, we might even be producing ketones as well. Now, even though we think of fatty acids as being the main fuel that's being sent to the liver in times of fasting, we can't forget about amino acids, which are released from our tissues, mostly our muscles, really, and they're sent via the bloodstream also to the liver. Now, once amino acids have arrived at the liver, the factory house, so to say, for energy production in times of fasting, remember that they can enter a diverse array of metabolic pathways. So I want to remind you, in our fed discussion, we talked about glucogenic and ketogenic amino acids. So in times of fasting, potentially these glucogenic amino acids can contribute to these precursors of gluconeogenesis and help support the production of glucose in times of fasting. Now, those that become intermediates of the Krebs cycle might potentially also contribute to the production of some ATP in the cell, but I want to remind you of the big picture that only about 10 to 15% of our total energy production is supplied by amino acids. So we really still think about fatty acids comprising the bulk of ATP production inside of our body. But these amino acids are clearly important for providing those carbon backbones to support glucose synthesis. And of course, these ketogenic amino acids could also potentially contribute to the synthesis of acetyl-CoA and subsequently ketones. So remember that the whole purpose of ketone synthesis was to try and preserve the degradation of protein in our muscles so that we could switch to a more kind of sustainable fuel based on the immense influx of fatty acids that we were getting into the liver. So really this acetyl-CoA that contributes to ketone synthesis, we think about as largely coming from these fatty acids. So I'll go ahead and kind of write this double arrow in to remind us of that fact. And I want to go ahead and scroll down and actually mention one unique thing about the catabolism of amino acids. Recall the basic structure of an amino acid, and I'll go ahead and draw the structure of an amino acid at physiological pH. So at physiological pH, we know that we have this carboxylate anion, and we have this carbon here attached to an amino group that's protonated. And we also have some type of functional group, which we usually abbreviate as R, which makes the identity of all these amino acids unique, and then have physiological pH. So at physiological pH, we know that we have this carboxylate anion, and we have this carbon here attached to an amino group that's protonated. And we also have some type of functional group, which we usually abbreviate as R, which makes the identity of all these amino acids unique, and then we can't forget this extra hydrogen here. So that's the basic structure of an amino acid. And the point that I want to highlight here is that something that's unique to the breakdown of proteins that we haven't run into in the breakdown of fatty acids or glucose is the presence of this nitrogen in this amine group right here. Now, notably, I did not mention that this amine group was contributing in any way to these precursor molecules that we talked above with regard to the breakdown of amino acids. And specifically, if you remember, I used the term carbon backbone of amino acids to refer to this part right here that was being converted to all of these precursor molecules. And indeed, generally, the first step involved in the catabolism of amino acids, or the breakdown of amino acids, is something called a transamination step, in which the amine group on this amino acid is transferred to another molecule for eventual excretion by the amino acid nation step, in which the amine group on this amino acid is transferred to another molecule for eventual excretion by the body. And that, of course, frees up the carbon backbone to contribute to the rest of these metabolic pathways. And so ultimately, this becomes something called an alpha keto acid. And it's called an alpha keto acid because of what its structure looks like. So it ends up looking something like this. It obtains a ketone group here, and of course, it's still attached to its R group. So it's alpha because it refers to this alpha carbon relative to this carboxylate ion, and it's a keto because it's a ketone, and it's an acid because it's attached to this carboxylic acid functional group here. So alpha keto acid, and this is the carbon backbone that can contribute to all of those metabolic pathways. And now, even though I'm only going to touch the acid, and it's called an alpha keto acid because of what its structure looks like. So it ends up looking something like this. It obtains a ketone group here, and of course, it's still attached to its R group. So it refers to this alpha carbon relative to this carboxylate ion, and it's a keto because it's a ketone, and it's an acid because it's attached to this carboxylic acid functional group here. So alpha keto acid, and this is the carbon backbone that can contribute to all of those metabolic pathways. And now, even though I'm only going to touch the surface of this, there's one last thing I do want to mention, that the common acceptor for this amine group, the common molecule that accepts this amine group from amino acids, is a molecule called alpha keto blue that accepts this amine group from amino acids. It's a molecule called alpha keto glutarate. And this might ring a bell because it is an intermediate in the Krebs cycle. And when it accepts this amine group, it becomes a molecule of the amino acid glutamate. And then finally, what the glutamate does is that once it reaches the liver, because the liver happens to have the right types of enzymes for this next process, it can donate this amine group in the form of ammonia, which is NH3. And I'll remind you that this, of course, is in equilibrium with ammonium and H4+. It will donate this as ammonia, and this will enter something called the urea cycle inside of the liver, where this ammonia is converted to a molecule of urea. And of course, this urea is then excreted in your urine. So that's how our body is able to effectively use the carbon backbone of these amino acids and also essentially detoxify our body of this nitrogen-containing amine compound. And the reason I should mention why it's so important to eventually excrete this compound from our body is because ammonia is toxic at very high levels to our body. So we need some way to effectively rid it from our body, and this is how our body does it.
Bravo. All right, let's take a 10-minute, and I'll go through and add in all the lessons to the Anki deck. Do that, and yeah, and then we'll go ahead and read high yield, and then we'll do the questions that I didn't answer before. And once the questions are done, we can go ahead and read the book. Read the book, Falcon book, and move forward. Yep. Cool. Let's take a break. 10 minutes. Yeah, so I'll start back from where I hadn't answered the questions. Maybe the ring. I think I had ring, although maybe the second one productive. Okay, I'll be back. Yep. Cool. Hey guys, I need to get some stuff from outside, and and then I want to make some eggs. So I'll be back. All right, I'm back. Sorry about that. I had to do a couple of things. 6:30. Good. Feeling good. Or feeling okay, I guess. Food. On my last meal of the day, actually. I can't be eating all that much. Oh. Here's what I did. It's just eggs. Pretty good. Pretty good. right. Yeah. Okay. It's out. Oh, I see. Um, yeah. Seriously? I can't even see the bottom. It's funny. Oh my goodness. I don't think we'll do. Bioenergetics and regulation. Oh wait, this is Kaplan. I must wonder for a reason, right? You. So that's the mosaic contraction already. So that's muscular and skeletal. Looks like they're together. That's it. Wait, did I miss something there? No, I don't think so. Cool. Very cool. What's that? A behavioral biochemistry. Yep. Cool. Okay. Nephron. Yeah, should have. This was a while ago. Why are those? reduction in oxidation reactions. That's interesting. Let me see. Oh, must be somewhere in somewhere actually. Yes. Okay. Oh. Yeah, so physical processes. Cool. Then it's not. I guess some of these is okay, right? I mean, it must have been any different reason. Take some things out. That's fine.
why it's not. Okay.
Really quiet. Or maybe. You say it just keeps going or what? Okay, that's okay. Process. RD. Let's take 10 minutes and then we'll read the high yield guide. And then, I guess I'll read the book. That's what I said I was going to do anyways. All right, let's take a 10 minute. Okay. I have to take out the trash, guys, so I'll be right back. I use the restroom. Yep. Let's read. You can see it. Hey, Chucky. How's it going, buddy? Yeah. Hmm. Oh. Oh wait, what? Oh, that's strange. Okay. Yeah. Mm. Oh, yeah, that makes sense. The long one, huh? And I'm just gonna basically keep going all the way down until we get sliding filament theory. Gotcha. Yeah, that's what I thought. Oh, I see him. Yeah. Yeah, I'm not sure. See? Yeah. Mhm. Mhm. Yeah. All right, glycolysis. All right, let's take a 5-minute. I think, yeah. Yeah, so it's glycolysis, Krebs, and ETC, and also phosphorylation. Cool. Yeah. Cool. Yeah. Because I have the thing, so and then this one. And then I guess GenChem through this. GenChem will be through. I mean, I guess it's just the same thing. Just keep. I think I'll just keep doing that. Yeah, because I just don't have enough time. Okay. Oh yeah, 5 minutes. Sorry. Yeah, give me 5 minutes. I'll be back.
strange. Alright, let me get some milk. I'll be back.
Yeah, tomorrow's Friday. We finish up with this, and as soon as I finish the book, I'll do Yusuf, and then Saturday is the 21st, and then Saturday I'll do the physio class when I'm done with Yusuf, and then yeah, soon Sunday will be all day physio. If I need it, if I don't, then we go ahead and move on. Oh man, seriously? Let's put some milk or something. Oh, that's good. Oh, yeah, nice. Yeah, I can see the guys coming off. All different types of sugars. Okay, cool. Oh, damn. Oh, I see what he means. Oh, gotcha. That's pretty nice. Where's my other one? left to right, down, up, angle. Yeah. So he's saying, what if we flip it? H, O, H, O, H, 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 O, H, C, H, yeah. What if we flip that always, making the addition? Left, right, left, left. And then dagger. Gotcha. Sure. Okay. Wait, okay. Focus on a head direction. Down, up, down, down, down. Excuse me. Oh, okay, cool. Batters up. Batters up. This is for animals. Animals, animal, carbon. Batters up. Batters up. Batters up. Batters up. Check out this picture. Yeah. Okay, I see it. steering. Don't worry about the name. Diasters. Dun, 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 Alright, so that was an ambulance. Is it shift lock? Alright, enantiomers. And then anomers. Enantiomers, anomers, down, up, down, down, up, down. Oh, wait. It's still the same, actually. It's still down, up, down. Yeah, down, up, down. Ladders up. Up, beta, glucose. And then alcohol group, alpha. Okay. Alright, what about diastereomers? The non-aromatic carbon. Is epimer of glucose. Glucose. Right. must have already been broken down into glucose. Oh, okay. Oh. Yeah. Right. Okay. Yeah. It's completely different. What? in the product is split from the molecule to two. Girls can't find food. Girls can't find food. Gentlemen, time, girls. Gentlemen, time, girls. Where's the G? Girls. Okay. Girls can't find food. Gentlemen, gentlemen, time, girls. Boys prefer eating like pepperoni pizza. Oh, I see. I see. Okay, okay. Yeah. Oh. Yeah. Okay. Yeah. Most get flat food. Most get flat food. Most get flat food. Watch food. One grade bits lost like this rate. Ten steps. Yeah, I can see. Yeah, so have to start memorizing it. Gotta start memorizing it. This one's here. Gentleman Darwin. Gentleman Darwin. Girls. What are you doing there? I don't like when that kind of skips on the thing we're trying to learn. Oh, seriously? That's such a bummer. Oh well. It takes a long time. It's just a lot of that chat. It's just so slow. I'm just gonna use a little. I mean, it's kind of, it is what it is, whatever. Fructose, 6 full pyruvate. I've been missing almost every other. Pyruvate kinase. Okay, and what else was there? The names of the substrate. Which steps produce FADH, NADH, CO2, or GTP? Excuse me. Okay. Alright, what we got? What do we got? What do we got? Alright. Irreversible steps. Irreversible steps. Alright. Occurs in a cytosol, glucose splits into two pyruvates. Names of each product. Sure. Two. I mean, that only occurs once, right? Step 4. At least, I mean, that's from what I'm seeing. I'm not seeing it anywhere else. When ATP is required. Required in stage 1. Required in stage 1. Produced in stage 3. And so is NADH. And ADH is also produced in stage 3. Not very much. What the heck? Well, I guess for this part, yeah. Sure. Low yield, don't have to know structure, don't have to know what the enzymes look like. What the structures look like. Enzyme irreversible steps. Save for glucose. Save for glucose and pyruvate. Save for glucose and pyruvate. Oh, save it for glucose and pyruvate. Sure. Pyruvate. Show me pyruvate. No. Just show me pyruvate, man. Pyruvate molecule of pyruvate. Seriously? Seriously? Oh, because I did videos. I'm sorry about that. Okay. You. Yeah. So I love to memorize pyruvate. That's okay. It's not that bad. Pyruvate. Something like that. So, I could do the other one. So I don't think they're gonna do this one. I doubt it. They usually never do. So let's do CH3, okay. Pyruvate. Pyruvate. Alright. Okay. And reversible steps. What more does it look like? Save it for at the end of now, so I'm focusing. Whether the respiration is anaerobic. Right. It's a triple anaerobic. Yeah, it's no oxygen. There's no oxygen. There's no oxygen. Yeah, which is here. Okay. On the next page is a visual. I think it's here. Anyways. Right. Right. Okay. Cardiac arrest or stroke. There's suddenly no oxygen, ETC stops, cells die, and oxygen is restored quickly. That makes sense. Okay, cool. All right. What else was this? What else? What else was there? Oh, yeah. My other question was. Uh, I'm sorry. Right. Oh, I see. Right. Okay. Yeah. Mm-hmm. Gotcha. Mm. Yeah, I gotcha. Okay. Okay. All right. All right. So, well, before we can even begin, I have to be able to, um, when it is produced on can't just say it was 3D. Sure. Shoot. We didn't see the answers, right? Oh, the enzymes are kind of like some stuff. Oh my goodness. Like, yeah. Maybe, that's fine. Which one? Maybe this one is. Anyways, okay, roll through. And then we'll just practice this for a little bit. And then if I feel like I have it, I'll go to sleep. We'll pick this up tomorrow and do the rest, and that's it. There's still 3 more kind of chapters in here to do. Yeah, I'll just go with it, I guess. Yeah, maybe tomorrow. I'll read it so we can read what the next thing I have to do. I still have to do the other ones. Oh man. Alrighty. Get back here. No, you come back. Alright, let's start. I don't know. Say it takes more than 7 times to memorize something. So we'll be doing it. We'll be doing it for a while. Okay, I'm gonna do good guys feel fabulous, doing good, but prefer perfect peach pie. Good guys feel fabulous, doing good, but prefer perfect peach pie. All right. Let me memorize that for a second. Good guys feel fabulous, doing good, but prefer perfect peach pie. Good guys feel fabulous, doing good, but prefer perfect peach pie. Good, but prefer perfect peach pie. Okay. I think I need to know. Then I need also, then I need. Okay. Okay. Yeah. Yeah, makes sense. High what? Something people. Okay, yeah, let's try. Sure. Um, sure. Okay, good. Good guys. Good guys. Fructose and supplies also doing hydroxyacetone. Phosphate. Glyceraldehyde through phosphate. One through five phosphate. One three phosphate. Sorry. Actually, I think I just do 1,3 BPG. 1,3 BPG. BPG. Actually, if I can chew it right now, no, I wouldn't know. It's just gonna sound like this one's fine. Good thoughts. Just I prefer perfect peach pie. I wonder if there's an easier way. There probably is, right? Sure. Honey Mary's glycolysis forever.
have to say it's wrong. Never forget how to be so. Just get rid of it. It's great. Hello and welcome to Med Simplified. In this video, we will learn about an awesome method to remember the steps and enzymes of
Yeah, but you
glycolysis.
have them all. Oh, you. Let me see. Have to make sure, you know. Because sometimes they're like always different. What happened? Dihydroxyacetylphosphate, glyceraldehyde phosphates, three phosphates. Okay, yeah. 3 phosphoglycerates, 3 phosphoglycerates, 2 phosphoglycerates, phosphophenylpyruvate, and pyruvate. That's right.
Simplify.
Are the intermediate strands? Are the other strands? I can't really see. I think so.
It's a method to remember the steps and enzymes of glycolysis in a simplified way. I found biochemistry extremely difficult to memorize when I was in college. Every pathway that I used to study, I used to spend hours on an end to remember that, and all of that was gone in a week. Eventually, you get to know about the awesome technique of using mnemonics as a memory aid, and I discovered some mnemonics to help me remember this complex pathway of glycolysis. Now, obviously, before beginning, I hope all of you have seen the videos of glycolysis on our channel. Well, if you haven't, make sure to do that so you have a better understanding of this topic. The link to the videos about glycolysis and other topics of carbohydrate metabolism are in the description below. So glycolysis is the initial pathway of metabolism of carbohydrates. Through the pathway of glycolysis, glucose, which is a 6-carbon compound, is broken down into 2 molecules of pyruvate, which is a 3-carbon compound. This is why this is known as a lysis or breakdown, since glucose is being broken down into smaller parts that make the reactions possible. Now, to remember glycolysis in a better way, you first need to remember all the intermediate products that are formed in the pathway of glycolysis. And then we will try to memorize about the various enzymes that are involved in the pathway
I
at
really need to know all of them.
different steps. So first, let's memorize all the intermediate molecules of glycolysis with a simple mnemonic. And to memorize this mnemonic first, you will listen to one of the most bizarre stories you have ever heard. And I guess that is probably a good thing, as that makes sure you won't forget it for a lifetime.
Okay, let's see.
So listen, there are 2 types of boys. One are good guys and the other are gross guys. The gross guys, they favor big butts, and the good ones, they prefer pretty girls in pink pajamas. Okay? Now,
Okay, for girls in pink pajamas.
you may ask, what does all of this has to do with the breakdown of glucose? Well, this story actually brings us to our first mnemonic. Gross guys favor big butts, but good boys prefer pretty girls in pink pajamas.
Girls, guys? Okay, it's a different mnemonic, I guess. Well, let's just see what happens.
And this mnemonic is based on the following substances. The G in gross stands for glucose, which is the first molecule used in glycolysis. Again, we have one more G that stands for glucose 6-phosphate, which is the next molecule in this pathway. The F in the word favor stands for fructose 6-phosphate. In the next reaction, another phosphate molecule gets attached to fructose 6-phosphate to make it a fructose 1,6-bisphosphate. Since it is a bisphosphate, now the word for it in the mnemonic starts with B, that is big butts. Next 2 words we have in the mnemonic are good boy.
Sorry, I'll change it. Girls, guys. He was saying, like, I'm gonna tell you a story. Wasn't really a story. guy's favorite. No, wait. This guy's favorite house. Good boys.
Is. The G in good stands for glyceraldehyde 3-phosphate, which is produced when fructose 1,6 bisphosphate is split into two molecules. One is glyceraldehyde 3-phosphate, and the second one is dihydroxyacetone phosphate. Only the glyceraldehyde 3-phosphate can move forward with the pathway of glycolysis and not the dihydroxyacetone phosphate. This is why only glyceraldehyde 3-phosphate is included in the mnemonic. The next word is boys, which stands
Yes. That's fine. Okay.
for bisphosphoglycerate. More particularly 1,3-bisphosphoglycerate. So the last line in the mnemonic was good boys prefer pretty girls in pink pajamas. And here you can see the next few molecules that come in the pathway of glycolysis. The P in prefer stands for 3-phosphoglycerate, which is a product formed when we remove one phosphate from 1,3-bisphosphoglycerate. The next P in pretty stands for 2-phosphoglycerate, the product formed as a result of transfer of one phosphate group from the third position to the second position. The P in pink stands for phosphoenol pyruvate, and the P in pajamas stands for pyruvate, which is the last molecule generated in the pathway of glycolysis. So this was a memory trick to remember all the intermediate molecules that form in the pathway of glycolysis. Now you also need to remember all the enzymes that are involved in these reactions. And trust me, when I was back in college, I found it extremely hard to remember the enzymes of glycolysis and
Um. Yeah, I need to memorize the enzymes too because then I won't know. I won't know which ones are the irreversible ones because I need to. Yeah, I need to know the whole thing. This is true. This is true.
Krebs cycle until I found some good mnemonics, of course. And for this, first listen to this small stupid story about a girl named
Girl named Geico. Oh my god.
Wow, our cats just love great customer service.
Helen. So this is
That's funny.
Helen, and she's a 15-year-old cute little girl living in her home. Just in front, and for this, first listen to this small stupid story about a girl named Helen. So this is Helen, And she's a 15-year-old cute little girl living in her home. Just in front of Helen's home are training grounds for players where they do their exercises and play and all that. Now, Helen used to sit along the training grounds and paint pictures all day long, and she used to pray that people enjoyed her paintings. And this brings us to our mnemonic that will help us to remember all the enzymes of glycolysis. Helen paints pictures along the training